Good afternoon, everyone. Thank you for coming. Thank you very much for coming. I want to welcome everyone to our 8th annual Heinz C. Prechter Lectureship. We began this lecture around, this is the 8th one, obviously 8 years ago, wherein we invite some of our collaborators, individuals around the country who are doing research in bipolar disorder and with whom we've interacted with, collaborated with, and want to continue collaborating with. And we're excited to have Dr. Kathleen Merikangas come here to talk to us this year. But before I introduce Dr. Merikangas, I'm going to invite Mrs. Prechter to come up and say a few words. And I want to just note that this has been a particularly good year for the Prechter Bipolar Research Fund. We've had a number of very influential publications. We've had, I think, around 10 or 15 publications, or in that between 10 and 15 publications so far this year. 10, I know, in the first quarter. We're into the third quarter already, so we're making a difference, and the difference is manifold. It's because of the efforts of our participants, the people with bipolar disorder that participate in our study. It's because of the information that we're getting out there. It's because that there's enthusiasm to support this research. There's enthusiasm at every level. Now, in introducing Wally Prechter, there are many things that come to mind, but I just want to point out a couple of very interesting things. Now, she's just been awarded, given an award with the premiere night at the Detroit Public Television for her work in bipolar disorder and mood disorder and psychiatric illnesses. Wally has received numerous public and awards for her work, both at the level of philanthropy, at the level of reaching out, and for her level of work and in the community, getting the word out about mental disorders, about bipolar disorder, and about the need to emphasize research, education, and enhancing all services related to mood disorders. So we've been working with Wally for many years. She started the fund 12, 13 years ago and moved it to Michigan probably around 9 years ago, every year, in 2004, 10 years ago. The years catch up with me. And we're very proud of what we've accomplished today, and I just want to invite Wally to come up and say a few words to kick off this Heinz C. Prechter Lectureship. Thank you very much, Wally.
Thank you, Dr. McInnis. I've decided I will not accept any more honors, but donations I will accept. And I've told my assistant, Leslie, that, and she knows this is what we say from now on. No more honors. It's too strenuous, and it's just, I don't want the focus on me. I truly don't. I would rather have it on people who have the illness and what we can do for them and what the fund will accomplish in the long run. That's really, truly what I want. I want to welcome all of you to the 8th Annual Prechter Bipolar Lecture, and I also want to thank our sponsors. They are Comerica Bank, Dearborn Sausage Company, Fritz Enterprises, Holbrooks Roofing, and Scott Snow Financial Advisors. It's very important that we have these people who give us support each year. We initiated these lectures in 2007. Each year they bring distinguished researchers to our university to give talks on bipolar genetics and research. They also spend time collaborating with our wonderful team under Dr. McInnis. I'm very proud of what has been accomplished since I first created the fund in 2001. Today, we are the largest research fund of its kind in the nation. We are preparing for a $25 million endowment campaign. We are poised for success, and I believe we are able to find the answers we're looking for. Again, thank you all for being here, and welcome.
Thank you very much. This is working out. Thank you very much. Now, it gives me tremendous pleasure to introduce my friend and colleague, Dr. Kathleen Merikangas. Dr. Merikangas received her bachelor's from Notre Dame University. She went on to University of Pittsburgh to do her Ph.D. in epidemiology, focusing on chronic diseases and the epidemiology of chronic diseases going forward over time. She subsequently located to Yale University, where she spent a good amount of her career in epidemiological research. I remember reading her papers in the mid-1980s, focusing on patterns of these illnesses in families and individuals. And the paper that I remember the most was the paper on assortative mating. And we read about that. We considered that and the environmental influences as how, individuals with depression, with bipolar disorder met and sort of were attracted to each other. So she's had tremendous influence in the field over the years. She's worked with some of the or trained with and worked with some of other leading figures in the world of research. Ries, who has been extraordinarily influential in the genetic analysis research. Kathleen and Neil published a paper in 1996 that really influenced how genetic research was done for the past 20 years. She relocated to the National Institutes of Mental Health in 2002 because she was interested in chronic disorders in general, not just one particular illness. And so she saw that as an opportunity to be able to study many diseases and interact with a host of individuals. So Kathleen works with individuals around the world. She is a very popular invitee to come to present at conferences. She collaborates with ever so many people, and is just a wonderful person and collaborator in every way. So it gives me great pleasure to introduce Dr. Kathleen Merikangas. And now I got to figure out how to get the... Now, do you want the lavalier or do you want this? Does this one work?
If I speak, is it on? Okay, so it's good. Okay. Okay. Thank you very much. It's really an honor and privilege to be here to present this lecture. I was here about 7 years ago when you first opened the center. And this morning, meetings with people who are working in your center that Dr. Graydon has established, along with the support of many other scientists and Dr. McInnis, it's remarkable how much stimulating research you have going on here in many different areas, and much of which is unique, I think, to what's going on all over the world. So I think you need to be congratulated for a wonderful accomplishment. And I've spoken with Dr. Graydon today. We need to build an infrastructure to study depression across the world because it's still one of the #1 health problems that we confront. So in my comments today, I'm going to be talking about my disciplinary field of epidemiology, and the title is Deconstructing Bipolar Disorder from the Population to the Clinic. Now, usually it should be from the clinic to the population. However, we've made so many observations in our population studies that I think will have major value in our treatment and follow-up of patients with bipolar. So I think it does go in both directions. So this is my disclosure. Our work is funded by the National Institute of Mental Health. We're in the intramural research program where I've been there, as Dr. McInnis mentioned, for the last decade. I moved there from Yale because I saw an opportunity for us to collect population cohorts and to work with experts across multiple disciplines who could provide insight and expertise into our understanding of the broad range of manifestations of bipolar disorder, as I'll tell you in a few moments. So this is the latest information from the World Health Organization that shows disability-adjusted life years attributable to mental disorders. It was published in 2014. And what this shows us, we never did end up with a pointer, right? But what this shows us is that if you look at the light blue, that is the DALYs attributable to major depression. So 40% of the disability-adjusted life years attributable to neuropsychiatric disorders are attributable to major depression. And bipolar depression, or bipolar disorder, has equivalent disability to schizophrenia, drug abuse, and alcoholism. So all of the major disorders that we study have equivalent disability, and if we couple that with the depression from which most people with mania and bipolar disorder suffer. It speaks to about half of the impact of this condition in the world. Now, in my comments today, first I'd like to talk about the contributions of epidemiology to our understanding of bipolar disorder. During the past decades, we've devoted substantial effort to trying to understand the manifestations of bipolar disorder in the general population. And I'd like to summarize some of the major findings from that and how we've incorporated those findings in our studies to understand the pathogenesis of bipolar disorder. Second, I'd like to summarize the results, the initial results of a very large-scale family study that we've collected at the National Institute of Mental Health through the local community, through Bethesda, the Maryland region, where we've enrolled a community-based cohort who we can follow and study intensively over time to address the questions that have to do with the core features and correlates of bipolar disorder. And finally, talk about some of the recent advances in the application of mobile technologies where we can begin to understand such constructs as mood regulation and the interrelationship of the homeostatic mechanisms that underlie bipolar disorder, namely sleep, activity, and mood states. So let me begin to describe what we've learned about epidemiology of mental disorders in the last couple of decades. This slide shows the sites of the World Mental Health Initiative that was conducted by Harvard University and the Institute of Social Research at the University of Michigan, where this study was administered in now 25 countries across the world. The flagship study was the National Comorbidity Survey Replication that was developed by Ronald Kessler in 1990, and then I became a co-investigator with him in 2000, where we did investigation of adult mental disorders in a representative sample of US adults. So this work has taught us a number of principles about bipolar disorder that's led us to reconceptualize how we think about bipolar disorder as we've moved to the population from the clinic. So this slide is from a publication that shows the age of onset curves, and it shows first, if we look at the blue, that's bipolar 1 disorder, the red is bipolar 2 disorder, and the green is the bipolar spectrum. So the two major lessons that these studies have taught us is one, that bipolar disorder is much more common than we previously believed. If we look at people who hover just below the threshold of disorder, they don't meet full criteria, but they have the symptoms of bipolar disorder. They may not last as long, they may not be as impairing. But when we start to look at people who have these symptoms, and they continue, they recur across the lifetime, the rates of bipolar disorder actually triple or quadruple. So that when we look at what is known as the spectrum concept of bipolar disorder, which was operationalized by my mentor, Professor Jules Angst in Switzerland, we see that about 4% of the general population will suffer from bipolar disorder across their lifetime. So the first observation then is that we need to look at the full spectrum of manifestations of bipolar disorder and mood disorders in the population. And second, we can see here that the onset of bipolar disorder is earlier than previously believed. So what we see is the onset starts to emerge in early adolescence. And as we go across the age span, it basically tapers off by about age 50. So we might have some shifts from major depression to people developing manic episodes later on. But I think the important message from this is we need to be looking earlier in the lifespan to examine the earlier manifestations of these conditions. The second major lesson from this work is that comorbidity with other conditions, both mental and physical, is the rule rather than the exception. So if this is a population-based sample, we see here that 70% of people with bipolar 1 disorder have suffered from 3 or more other mental disorders in their lifetime. It may not be concurrent, but many people start out with anxiety disorders in childhood, attention deficit disorder, eating disorders, and certainly substance abuse, which is one of the major complications of bipolar disorder. When we see these people in the clinic, we oftentimes are not aware that they originally suffered from manic episodes that led them to use substances, either drugs or alcohol, to help to treat the agitation that accompanies manic episodes. So this we've shown again and again in prospective studies in Europe and the US that this is one of the major opportunities for prevention and early intervention. Because once people develop substance dependence, it is a very difficult challenge that people suffer from for the rest of their lives. And we may not then understand the underlying pathogenesis of the conditions that are leading them to develop substance abuse. So I don't want to show you the slides now for the sake of time, but the other pattern of comorbidity that is pervasive is comorbidity with physical conditions, particularly cardiovascular disease, asthma, allergies, migraine, pain syndromes. We found that 50% of people in the general population who have bipolar disorder suffer from another major medical condition. Some of these medical conditions predate the onset of bipolar disorders, and others may be complications of bipolar disorder, such as obesity, metabolic syndrome, which may be secondary to the drug treatment, or they may be part of the pathogenesis of the stress system related to bipolar disorder. My mentor, Jules Angst, has recently published data from a 50-year follow-up of patients with bipolar disorder in the Burghölzli Hospital in Switzerland. And his data showed that cardiovascular mortality is specifically associated with mania and not with depression. Most of us who have been studying this have been looking at depression and cardiovascular disease, but his data show that people with episodes of mania across the lifetime are more likely to die from cardiovascular disease than people with bipolar 2 disorder or people with depression. He's also looked at suicide, and deaths from suicide are primarily attributable to the major depressive episodes associated with bipolar disorder. And particularly the bipolar 2 subtype has an insidious increase in the risk of suicide across the lifespan. So this speaks to the importance of comprehensive evaluation of people with bipolar disorder, that when we have specialty clinics, we may miss the importance of comorbid physical and mental disorders that may actually lead to disability, impairment, and so forth, that persist after our ability to treat the acute episodes and the syndromic manifestations of bipolar disorder. Now, people with comorbid disorders tend to have much greater severity. So first, comorbidity is the major explanation for differences between who we see in clinics and who we see in the population. There are numerous people in the general population who never seek treatment for bipolar disorder because they don't have the complications of bipolar disorder, or they're not recognized for comorbid mental or medical disorders. So people with comorbid disorders are much more likely to come to your clinics, to our clinical attention, and we already see this emerging in adolescents. So the people we see have a much poorer course when we do follow-up studies. Literally all of our follow-up studies in the US are based on clinical samples. So actually, there is a much more optimistic outcome for some people in the general population who may not suffer from the comorbid conditions that lead to disability and impairment. So I won't go through all of these points, but functional impairment is particularly associated with comorbid disorders. And if we could cast a wider net, treat the full syndrome instead of just focusing on the symptoms of bipolar, we might be able to improve our ability to implement change in people's lives and their disability over the lifespan. So, this illustrates what those of us in epidemiology are very interested in. And people in genetics were the first to characterize differences between who comes to the clinics and who is seen in the population. Because recessive traits never appear until you have 2 copies. So those are clinically ascertained because people develop the disease. The same is true in psychiatry, where the majority of people who are seen in clinical settings are much more severe than those people who we see in the community. In doing our study in the greater Washington area, I've been absolutely amazed about how many people suffer from bipolar disorder who it hasn't been recognized nor treated. And if they are going to get treatment, they are seen in primary care. They're treated with SSRIs for depressive episodes. And if they aren't experts in mental health, oftentimes the mania, the other problems from which people suffer are not recognized. And then they have poorer outcome. This gap has been growing over the last decade. Fewer and fewer people with bipolar disorder, and this goes down to adolescents, are seen by specialty mental health care. Now we see children are much more likely to be treated by pediatricians and adults by primary care physicians. So it's very difficult to bring people to mental health clinics because of some of the restrictions in the health policy and people's ability to get to see mental health professionals. We could offset, I would suspect, 30% of suicide attempts and people desisting to take their medicines because they're not seen in specialty mental health clinics where we deal with the full spectrum of the disorder and people's contexts. So I think there needs to be a major shift in bringing people back into mental health care. And that requires us training many more people, particularly in child psychiatry, where there's a tremendous tremendous shortage of people who have been trained in particularly treatment of mood disorders and treatment of mental disorders. So to go forward to talk about moving down the age spectrum to adolescence, in the last couple of slides for the epidemiology component of my talk, I wanted to talk about our study of adolescence in the United States. This is the first national community study of mental disorders of youth ages 13 to 18 across the U.S. It's a representative sample of the U.S. Again, the University of Michigan Institute for Social Research actually conducted this study. When I was at Yale University, we worked together with David Offord from Toronto and Ron Kessler from Harvard to validate the instruments to move down to age 13. Now, we didn't go younger than that because there's a tremendous disagreement about how we measure these syndromes earlier in the lifespan. But I'll show you in a moment. The manifestations of bipolar disorder and mania that emerge in adolescents are very similar to what we see in adults. So we're beginning to see that this is really the adult form that most of us who have worked in the treatment settings have seen in adults with this condition. So by moving down the lifespan, we're beginning to see the emergence of these conditions. So first, this slide shows, and I think this was just remarkable to us, if you look at the left, we found that only 50% of youth who met criteria for depression or mania with severe impairment were being treated at all for those symptoms. So only half of them were being treated. About 13% were being treated for something else, usually attention deficit disorder or anxiety disorders. And then about 40% were not being treated at all. So then if we look at what they were identified for, I don't really have time to pursue this, but mainly people were seen for depressive episodes. And hence, if they're seen in primary care settings, and people don't recognize mania, they don't recognize anxiety, they're not taught to look at the subtleties of agitation which accompanies mania, then oftentimes we really send them on the wrong path. That is not going to really help them recover from their illnesses and may indeed induce greater chronicity. This slide shows a statistical approach that we used called latent class analysis to look at the structure of symptoms of mania in adults and adolescents. So the top part of the slide shows adults from the general population, the National Comorbidity Survey, adults, and then adolescents from our National Comorbidity Survey Adolescent Supplement. So the first finding here, if you look at the red, that's high probability of all these symptoms we show here. These are the core symptoms of mania. So you have these red subclasses where people tend to have all the symptoms. So these are the kind of people who come in, you know they have mania, they have most of the symptoms, it's kind of undeniable. And then at the bottom, we have people who endorse having some manic symptoms, but they don't have a high probability of all the symptoms. So these are people who are less severe. They have manic symptoms, but may not meet full criteria. And in the middle, we have subgroups with different prominence of either the cognitive or the motor physical symptoms. Okay, so we see that in both adults and youth, the same patterns. So this suggests to us that the mania that we see emerging in adolescents ages 13 to 18 are very similar to what we see in adults. The second major finding from this slide, and this has actually led us to completely shift the way we think about bipolar disorder, was that increased activity is the major symptom of mania in both adults and children. Across all of these classes, the increase in activity was the cardinal symptom that characterized episodes of mania. So if you look at these, you'll also see increased speech, or decreased sleep, and so forth. But the activity just jumped out at us as one of the most important symptoms. And as we've started to explore this more widely in the population, and we're beginning to do this across the world with colleagues in Australia, Norway, and Switzerland, we found that if we ask about episodes where people have increased activity, and we don't ask about increased mood, self-esteem, and so forth, we are getting a much larger proportion of people who actually meet full criteria for bipolar disorder. And in fact, as I'll show you in the next slide, mood changes probably are not even a core feature of the condition. So this slide shows the rates of major depression among youth in our population-based study who suffered from one of these other conditions. So if we look to the left, youth with generalized anxiety, about half of them had an episode of major depression. PTSD was the same. Mania, it was lower, about 38%, and so forth. So what this suggested to us is that major depression is not uniquely coupled with mania. That major depression is associated with almost any of these disturbances. If you have disturbances of eating, if you have disturbances of fear system, if you have disturbances in cognition, major depression looks like it's common across all of these conditions. And in fact, major depression alone is extremely rare. So this led us to think, well, maybe in its emergence, maybe major depression isn't core to this condition at all. Maybe it's the mania that we should be focusing on. And maybe depression occurs later, and maybe that is what leads to the serious sequelae such as suicide attempts and so forth. But maybe it's not part of the core features of mania, or whatever the disorder is that we're studying. So, this is a summary of what I've just told you about what we've learned from population-based studies. I think we've learned a lot. This has led us to start thinking about these features, these characteristics of mania, and to start to apply these in the studies that we're doing in both clinical and population samples. So now to turn to the second section of my talk today, I wanted to briefly describe the study that we've done at NIMH. And the reason I went to the NIMH from Yale, I was pretty well established there, and I moved to the NIMH because I was advised by many people that, number one, in that era, I could not get a family study funded, an extensive family study funded, because That's when we shifted to the era of genome-wide association studies that Melvin had originally talked about. So everybody was doing larger and larger samples of cases and controls. But my argument always was, having started my career working with families of people with mental disorders, that we needed to study families. And even if we didn't find genes, it didn't matter, because it's related to families. We need to work with families. Children are at risk of bipolar disorder and many other disorders. Even though they only have 10% risk, the disturbances secondary to having a parent with bipolar disorder are extremely important. So that if we studied families, we could also help the families. We could begin to intervene and also learn a lot more about the contextual manifestations of bipolar disorder and help to educate families about how to maintain people on their medications, how to bring them in, how to treat them, and so forth. So the family study to me is still the most potent approach we have to understanding these conditions that really tend to run linearly in families. So what we've done is we've moved from these big population surveys, we've then looked at extended families of people with mood disorders. And what we did at the NIMH is we cast a very wide net. We did a community survey to recruit people to look at mood changes, health, behavior. So we didn't want to just go out and ask for people with bipolar disorder, major depression. So we recruited people with sleep problems, people with problems with anxiety. We've been very interested in migraine. So it was mainly a study of health and behavior. And then we supplemented the study with people with bipolar disorder and major depression, so we had sufficient numbers of people with these conditions. But in general, we did not want to use a clinical sample because of the biases, as I earlier mentioned, in people who come to the clinics for treatment. So if we're studying the comorbidity and how that runs in families, then you're really dealing with the tip of the iceberg. And we wanted a broader manifestation of these conditions. So that's what we did when we moved there. And then finally, in the last few minutes, I'll talk about how we studied biomarkers, biologic markers, and this doesn't just mean blood, and looking at blood tests. We wanted to look at the core features of bipolar disorder, but we wanted to study them in families, because the only alternative we have in bipolar disorder to studying biomarkers is to look at family members, many of whom do not have the full-fledged disease. They may not be on medications, and that's the only way we can really study these biomarkers without their being influenced by concomitant medications. So that's why by looking at families, this can give us a clue. Well, the genes run in families, so do cultural factors, but this could give us an index of what are the core features running in families. So our family study was started in 2002. It is ongoing, so I have a plus here. To date, we've recruited about 500 adult probands from the community and non-clinical settings. We have 1,200 relatives who we've brought into the NIH Clinical Center, just shown up in this corner. We bring people in, we do a full battery of assessments, as I'll show you in a moment. We don't exclude anybody by age. We've only gone down to age 7. I was at a meeting of SAMHSA a couple weeks ago, and they're saying, well, we know a lot about infants, so you really should be characterizing these kids as well. It's not an area that I'm familiar with, but we at least enumerate the children in the families, and then these people live all over the country and world, so we have to do telephone interviewing of most of these people, and then we've been very interested in hoping to collaborate with you, where we could send people here, you could send people to us for some of the measures that each of us specialize in. So that's one of the, I hope, one of the conversations we can have while I'm here and in follow-up. So what we did is we developed mainly instruments. I could also not get money to do instrument development. You're not going to get money to do this anymore. So what we did was we expanded the DIGS, which is the diagnostic interview you use here, to make it much more phenomenologic. This is after Jules Onks, my mentor, when he developed measures, he says, you Americans have it all wrong. You use these instruments that are tied to the DSM. So you ask people, have you had a change in mood? You have to have this many symptoms for 2 weeks or more, all day, every day. And basically, that's not the way people manifest these symptoms. People with mania particularly, it only lasts for a day. People have this for a day, but then it comes back. We don't even consider recurrence as a criterion in our diagnostic criteria for bipolar. People can have one episode and they're characterized as bipolar. But what you see in general population samples in Switzerland is it comes back. So it may only be a few hours, and then people start to have it again and again. So what we'd like to do is characterize the phenomenology. And only after we do that, then we look at duration, recurrence, severity, and so forth. So our interview is very much like what you used, but now for bipolar, the important change is we don't ask only about a mood change, because we're now convinced that we have to ask about activation. So we ask about changes in activity. And for that, we really are finding that we're identifying much more that's going on in the families and in the population. And the other thing I mentioned is we're doing the biologic measures in our families, and then we do a full neurologic workup and a cardiovascular workup. So we actually do Holters, we do full neuro workups, we do neuropsychological testing. Let me go on to that now. Oops. I don't have all the measures that we're doing, but we're trying to look at the physical characteristics, the physical disorders, sleep disorders and so forth, as well as mental disorders in our samples. Okay, so this slide shows the major findings. They were just published in Molecular Psychiatry this past year. And what we did was to investigate the relationship between mania and depression based on the observation in adolescence that mania and depression may not be uniquely coupled. So we wanted to know in families, what is the relationship between mania and depression? So first, we looked at mania in probands and mania in their relatives. And what we find, this is an odds ratio of 8. This means that the risk to relatives of people with mania is about 8-fold that of controls and people with major depression and so forth. When we look at the heritability, and that's the extent to which we see aggregation within families, it's 0.83. It's suggesting that mania is hugely familial. Now, because we don't have twins, this isn't genetic heritability, but this is really a high heritability estimate for mania. Then we looked at hypomania, and we found hypomania wasn't even significantly familial. And I think that's because it's very difficult to identify hypomania, because by its nature, hypomania, which characterizes bipolar II disorder, is a disorder that doesn't lead to impairment. So you have increased energy, you have increased activity, but you don't have impairment. So how's that a mental disorder? Well, now we've started to study this more intensively to understand what is hypomania, what characterizes bipolar II disorder. These people don't have full-blown mania, and I can comment on that later, but it turns out that this is very different from bipolar I, and it doesn't look like it's on the same spectrum. Third, we found that major depression is familial, about a 2-fold increased risk. We now have 15 controlled studies. This is what we get in every study, about a 2-fold elevated risk. So mania was much more familial than depression. But the big question, if we have a unitary dimension that goes from depression to mania, which was originally proposed by Jules Angst in Switzerland when he deconnected major depression from bipolar disorder. So if we have this underlying dimension from depression to mania, what we should see is that people with mania should have an increase in depression in their relatives, and people with depression should have an increase in mania. So when we looked at that, we found absolutely no evidence of cross-transmission. So this suggested to us that though mania and depression co-occur, and probably in the majority of people, probably everybody you see in a clinical setting, but it looks like the underlying pathogenesis is separate. And now we're beginning to think about mania, 2 dimensions. One, which is characterized high energy, high activity, and fatigue at the other end. Because when we've just started to look at the atypical subtype of depression, it's much more heritable than major depression, than just aggregate major depression. So with my colleagues in Australia, we're actually beginning to look at activation fatigue states as the core underlying feature of mania. and depression, going from depression from good mood to bad mood as a separate dimension that co-occur, but in terms of their underlying biologic underpinnings may indeed be distinct. So in summary, mania and depression appear to have distinct underlying pathogenesis despite the fact that they occur. We replicated this in an independent sample. I think this is extremely important because I questioned our data, but my colleagues in Switzerland who have a larger sample than we do replicated this almost to the same odds ratio, and because they had people who were more ill, they then looked at psychosis and found the same thing. A main effect for psychosis, irrespective of what we call it, schizoaffective manic, schizoaffective depressed, schizophrenia, psychotic depression, there's a main familial effect for psychosis. And indeed, that's how we treat people. If someone comes in with psychotic symptoms, we treat the psychosis. If they come in with mania, we try to treat the mania. If they have depression, we treat the depression. So I think this is beginning to tell us, look at these underlying dimensions. Maybe these disorders, as we think of them, may not be the phenotype that we should be studying. And we're also following up high-risk offspring. We have 200 at this point, and we're going to continue to follow them as long as we are able to do this. And we're beginning to do the biologic measures. So in the last part of my talk, just want to check my time, I'm good. So the last part of my talk, I wanted to talk about the biologic measures we're doing. And I know University of Michigan, your group is involved in the EMBARK initiative, which is looking at biomarkers. Some of the things we've talked about this morning that you're looking at, the speech patterns, the diet, I mean, the things you're looking at are extremely important. I think they're going to really help us because they're really looking at these core features. And if we can use family data on the one hand, or follow-up data to look how stable they are, how core are they to bipolar disorder, I think by using these two strategies together, that we may be able to understand what are the core features, where do we need to put our efforts to understand where we might intervene. So this slide shows all of the domains that we measured when we first started this study. Of course, it's beyond the scope of my talk to talk about all this at this point. Plus, we're just finishing this study, the first phase with the first 500 probands. We hope to continue to recruit probands. But we did all of these kinds of measures that at the time of the study had been demonstrated in a case control study to be specifically associated with bipolar disorder. So the most exciting findings for us that we have really jumped on, and I want to show you these initial results, were in biologic rhythms. So as I mentioned, we were interested in activity, so we started to analyze the data, and I think it's really made us rethink how we think about the underlying phenomenology of bipolar disorder. So, our idea that activity is a core feature of mania, of course, is not new. If you look at this quote by Emil Mendel, it is not the Mendel of genetics who we've all studied. Here's a neurologist and psychiatrist in Germany who described manic episodes. And remember, in Switzerland and Germany, they lived with patients. Jules Onks told me we lived at the hospital. And then we'd try new drugs. We'd see somebody, Bloiler, his mentor, would say, let's try clozapine on this person. It doesn't, the drugs we're using don't work. Let's try this. And then that's how many of these drugs got discovered. The monoamine oxidase inhibitors were discovered because they treated people with tuberculosis and they alleviated sad mood. So this is the kind of observation, they'd look at these people, they'd study patients for years. People would come in and out of the hospitals. So Mendel characterized mania as increased excitability of brain processes. And when you think about this, and we know the biology of excitability of brain processes, it could affect multiple domains. It could be your thoughts, it could be your energy, it could be your speech, it could be your movement. So when we saw this, and he said, mood change is not even a core feature. So that really is a very interesting observation that our data, you know, a couple hundred years later, 150 years later, are actually telling us that his observation may indeed have been accurate. So we use mobile technologies to investigate people's mood regulation. We've been very interested at the NIMH in suicidality, suicide attempts, and all the consequences that we see, particularly in bipolar disorder. So we were interested in looking at objective measures in vivo intensively, rather than as we do with questionnaires. We ask people, even in clinical trials, we see people weekly or biweekly, and we ask them about the last week. So here what we wanted to do is to look objectively at what happens to people's moods across the day. How does anxiety change across the day? What is the relationship between the mood states? So what we did was look at what the Larson mood circumplex, which are 7 emotional states, where you look at them on an analog scale from 1 to 7. So this is ecological momentary sampling. It's been widely used in many other fields, particularly drug abuse. I understand that you're doing some of this. There's a lot of work at Michigan looking at smoking cessation. So this is a well-established tool. It has been used to look at people with bipolar disorder as well. So what we did, this is, do people know what this is? This little device here? My summer students laugh at me when it was a Palm Pilot. Because we started this in 2002, and the students said, what's that? You know, they've never seen a Palm Pilot. Well, now we're, using droids. But basically, you have an electronic device, doesn't matter what it is, and people answer it 4 times a day. So we do the mood circumplex, we look at what they eat, we look at exercise and activity. In the morning, we ask them how they've slept, how long they slept, and then we can couple that with some measures, objective measures of sleep. We're also interested in stress, so we ask them about stressful life events across the day and so forth. This technology has infinite applications in health, and it's being widely used, so this isn't novel. What's novel in our study, there's only a few others who have coupled the daily diaries with actimetry. So that's the Philips Respironics Actiwatches. So we put those on people for 2 weeks. They wear the Actiwatches, and then we pair them to the electronic diary so that we can then look at activity and how is that related to their mood, how is it related to what they eat, how is it related to sleep. So when we look at all these systems that we call homeostasis, regulation of your temperature, of your mood, of your energy, of eating and so forth, of fear, all of these systems are really important, but most of us can only study 1 or 2 at a time. So this opens a lot of opportunities for us to look systematically to see, is it a disturbance in the regulation across these rhythms, or is there a core system that goes awry that leads to a shift in the others? Okay, so that's what we did. We've done this in about, at this point, 400 people, some of whom have bipolar 1, some bipolar 2, some with depression, some with anxiety, and then we have people who don't have any of those conditions. They are usually not in acute state. So we're now in the inpatient unit at the NIMH with Carlos Zarate. We are going to be looking at people in acute states, in and out of states, because it's not so easy to do when you have somebody who's, having major episodes of mania for them to attend to these syndromes. So let me just show you a couple of findings before I close. So this slide shows what we're measuring with our activity monitors. So if you look from midnight to midnight, we have 24 hours. These Actiwatches, and I'm wearing a Fitbit. Everybody seems to be wearing these things nowadays, but what you do is look at the amount of activity every minute of the day. So you look at the counts of activity from midnight to midnight. We do it for 2 weeks. It turns out weekends, getting a full weekend is extremely important, particularly with people with bipolar disorder, when they have less structure to their lives if they're working. So we do it for 2 weeks. And what we're looking at here is how is activity during the day related to sleep? Because we're interested in sleep and activity both as core features. So what we show here, if you look at the bar graph up there, yellow is high amount of sleep, blue is low amount of sleep. So to call your attention to the controls first, what we see is if you have more sleep, and this was kind of counterintuitive, more sleep is related to less activity throughout the day. If you look at the yellow, they have less activity. Everybody has this increase, through midday, and then it goes down again. And there's a lot of characteristics of these activity curves that I'm not showing you. This is being used in the National Longitudinal Study of Aging. Age is the biggest predictor of patterns of activity. So if you have one age group in your study, it really does not represent changes across the lifespan. As people get older, they have a shift from morningness to morningness and much less activity later in the day. So we have to characterize age and sex and all these things. So it's quite complicated analytically. But the finding here for bipolar was what was quite stunning to us, and that is people with bipolar 1 disorder have less activity like everybody else up until about noontime. But then people with bipolar who had more sleep have this shift where they have more activity later in the day. Now they tend to have that, but it is related to the amount of sleep they get. So if you just look at activity, you won't see this. And most of the studies just average activity across the day. So the second thing that we found that I'm not showing you is people with bipolar disorder, and this is when they are not necessarily in manic or depressive states. They have much greater variability of activity. So if we look at each day over 2 weeks, they are all over the place. They just don't seem to have rhythms that are stable, particularly on weekends. So variability, and this has been found, I would say, in 5 or 6 other studies that weren't controlled necessarily, but much greater variability. So this is averages, but if we look at variability, you just see that people with bipolar disorder they're quite variable. So my colleagues in Australia are trying to implement treatments, interventions, both behavioral and medication, that actually helps to stabilize the variability of these patterns. Okay, now to move on to the second finding, we looked at depressed mood throughout the day and how that was related to activity, and this is exactly what you would predict. So yellow is high sad mood. This is an analog scale from sad mood to good mood. You feel good or you feel sad, an analog scale. So those people who have the highest levels of sad mood have less activity throughout the day. Okay, and there were no interactions with bipolar 1 on this. It was just that there's a direct relationship between activity and mood. Okay, so this was the other finding. We have several others that we looked at other systems. I'm not going to show you energy, but energy turned out to be one of the most important domains. So now I want to move to the lagged effects that we looked at. So here, we look 4 times a day. So this is the morning. These are self-defined, by the way. We do this in kids in school. So they measure this at particular times of the day when it's convenient. So we don't do this at random. So we get better compliance if we know when people are having lunch or when they can take a break. So basically, if we look, this is when they get up in the morning, and we have a mood measure. We have an energy measure from 1 to 7. How energetic do you feel right now? It's psychic energy. And then activity is measured by the actual objective measure of the ActiWatch. So what we're interested in here, we want to look at the stability of these states, these domains across the day and across the weeks. So the biggest finding, of course, is the stability of these factors across the 2 weeks. Bipolars have less stability, they have more variability. But basically, the biggest predictor of energy today is going to be your energy level yesterday. Same with activity, and same with mood. But the more important question to us is not the stability or instability within these traits, but how are they related to each other? So what we did was what we call lagged associations, where we look at does energy in one period predict activity in the next. So we wanted to know if you have high energy, do you have an increase in activity? And then concomitantly, if you have an increase in activity, do you have an increase or decrease in sad mood? So that was our main question, and indeed, we found that energy predicts activity, activity predicts a diminution in sad mood, and we did not see directional effects from activity to energy. So increased activity does not lead to an increased energy, nor does less sad mood predict more So in summary, what we found is a unidirectional association such that what we believe, what is driving this cascade, is that energy is related to increased activity. Activity is related to a diminution of sad mood. There are interactions such that people with bipolar disorder have much tighter linkage between these systems. So if somebody with mania or bipolar disorder has disturbance, if they have a disturbance in sleep, they have much greater change in mood, much greater change in activity than the average person in the general population. So they're much more sensitive to perturbations in these systems. So if we are interested in treatment, if we want to treat depressed mood, maybe it is that we're focusing on mood, treatment of the mood. Maybe what we should be doing is treating the energy. Once people feel psychic energy, then they become more active. These trials of activity to change depressed mood have not been very successful today. Well, maybe it's the energy, maybe it's this feeling of fatigue that is really preventing people from benefiting from increased activity in mood. So this leads us, many people have commented on this, we presented this at the Maudsley last week, and some of the medications we used to use in the past actually have some stimulating effect, that people have more energy. Once they have energy, can we then change this whole cascade that leads to depressed mood? Okay, so finally, as I just noted, people with bipolar disorder have much tighter associations in these systems. This suggests to us, and this is a small sample that has to be replicated, but what this suggests to us is that we should be looking at these systems to see how they're co-regulated. We should look at energy and activity and mood states in people with bipolar, because indeed it looks like it's a circadian disturbance. It's a disturbance in the interaction of these systems. So that our future studies may want to intervene farther back in this pathway than trying to change the depressed mood, which may be alleviated if we're able to address the earlier manifestations of this cascade. So in summary, for the biomarkers, the circadian patterns and increased variability of activity is a key feature of mania that we think warrants further investigation. I had a tour of your sleep lab this morning. That you have a beautiful opportunity to help to shift some of these rhythms, if indeed these findings are replicated. I think the use of mobile technologies, which are now being extended to prevent suicide by some of my colleagues, where they actually look at changes in activity. One of the findings from the sleep field, the editor of Sleep told me that he sees it over and over again. Doesn't matter what the diagnosis is, but oftentimes before someone commits suicide, they are reported to have been up all night. They don't sleep for 12 hours, they're very agitated. If we could wear these devices and start to see these kinds of changes, we may be able to go in and intervene. It's possible. So now I think these tools can help us, because people aren't going to call us. That's what we'd love them to do, but it just doesn't happen because they're so hopeless. And finally, this unidirectional association, I think, should make us rethink what are the targets of our treatment. How should we treat these conditions? So, I wanted to end by saying it's very sad to me that U.S. is way behind in this kind of work. My colleagues in Australia, Ian Hickey and Pat McGorry, have been working with the health ministers of their country to build programs where young people can come in through primary care, even kids in schools. We talk about these things. They're trying to get me to go there to talk about this, where we can teach children about self-regulation, about sleep, about having regular, stable rhythms. So the question is, can we offset some of these diseases? Canada also has a healthcare system where we can bring in large people. We're beginning to do this in Toronto with the teams of people interested in depression there. And in the US, we lack a national database where we can track people. So these countries have the opportunity to track people over time. I think if we could build that, I talked to Dr. Grain this morning about doing that across centers. I think that would really help us to apply these technologies in the future. So I just wanted to thank everybody who's been involved in this work. These are my NIH collaborators to do this kind of collaborative work. It really requires experts from multiple disciplines. And I was told that the NIH would be somewhere where I could walk across the hall and talk to experts in cardiovascular disease, neurology, and that's partly true. I think at your university you have these kind of opportunities as well. These are my collaborators outside the NIH. There's a whole host of them. And then finally my lab at the NIMH. So thank you very much.
Thank you very much, Kathleen, for a very energetic talk. And I just want to open it up for comments and questions. Yes. Now, just from the technical, do we need how the recording of it has gone okay and has been picking up well? Okay, got a thumbs
For
up
anybody
there.
beyond
Do you want a microphone here to send around? Okay.
Thank you for a very stimulating talk. I have a few comments and questions about some of the more biological, phenomenological things, but I'll just start by saying that some of us who are clinicians tell our patients that exercise is as important to treatment as their antidepressant pill, and say even if you can just start with 5 steps, go outside, take 5 steps forward, 5 steps back, and then tomorrow you'll do 6, but that it's a critical
That's fantastic because the data from adults, from elderly people, there's now follow-up data that shows if they just get people to move, they have much less cognitive decline. So just getting up out of their chair, it's not the cardiovascular activity, it's not getting on treadmills, it's moving. So if we do that with people, particularly atypical subtype of depression, and you get them to move, you can actually relieve these. So that's wonderful that we do this in clinical practice.
And I just think it's a combination of the psychomotor system, whatever, that the more activity that you do, the more your cognition is going to speed up, so for the elderly too. So one thing was, I don't know where you place irritability is among your phenomenological symptoms, because it used to be in DSM-2 or 3 or one of the older ones that irritability was one of the mood symptoms. You didn't have to have depressed mood, but you could have irritability as a symptom of depression. Now, of course, irritability crosses over because people with mania also have irritability. But certainly, that I would wonder, where irritability fits in terms of activity and anxiety and some of the other.
We can look at that we haven't analyzed that yet, but our problem with irritability is that irritability pervades across all disorders. People with generalized anxiety probably have more irritability, people with depression. Now, irritability can become very core to mania as well. So I think it's important because it is a change in mood. And I just have to analyze the data to see where it fits with all this. But I agree we should be measuring it. I just don't know where to put it.
It's not exactly an affective symptom,
Right.
but it's a measure to me of increased excitability. The other things about steroid psychosis.
I will get back to some of your questions in a minute. If there are other questions that we have from the crowd, the gentleman in the back there, please. So we have
Can you define activity?
Yeah, activity, the way we are defining it here operationally is it's movement, the number of steps you take. So the ActiWatch measures change in the movement.
So sitting
that's really
here,
what
I
it
would
is.
have little activity.
Right.
Okay.
Well, you could be moving, actually, but it has to be walking because they put me on a
But if I
recliner.
sat in a conference for 2 to 3 hours, I wouldn't be active.
No, you're not
Okay,
active.
then the other question I have, this energy activity mood, is this unidimensional? Can increased activity increase energy?
So we did not see that. That's what's interesting. Increased activity, the people don't get an increase in subjective energy. So that was surprising because I was told that if you go out and become active, you become more energetic.
The activity was
Certainly
measured-
didn't show that.
The activity was measured by your little device.
Yes. It's a big device, unfortunately. It's ActiWatch, $1,200 each.
Thank you.
Could you give some examples of the more stimulating medications that you're suggesting using in bipolar illness?
Well, stimulants, but the monoamine oxidase inhibitors that when I was trained in Pittsburgh, The cocktail for bipolar disorder by John Himmelhoek was a combination of lithium and trinylcypromine or phenozine. They metabolize to amphetamine, and those have been shown in clinical trials to be more effective for the atypical subtype of depression. People don't really use them in the US, although a lot of experienced clinicians still are using them, including Carlos Zarate and people at the NIMH when they treat patients. And in McLean, they're still widely used. So they, and particularly in elderly people, they metabolize to amphetamine. But Jules Oxt and others in Switzerland actually give people some stimulating medications. They give them stimulants. They don't keep, I mean, you have to monitor it. You bring people in, you just send them out to stay on stimulants. But a lot of times they will use stimulants when they have somebody who has the atypical depression to try to get them out of it. And then they discontinue the stimulants after the acute episode goes away.
So this lady break out, so John and then the lady at the back there.
Kathleen, thank you. It's another tour de force from you. I love those. So several points. One is really that through the years, this question of where are we with chronobiological rhythm has been a big one. The phase shift, Anna Justice used to talk about rhythm and moods.
Yeah.
You know, and
Absolutely. There's a whole history.
I know. And in the framework of that, earlier studies came up with some of the bipolar people having what they called hyperphagia and hypersomnia. And so they slept more. The idea being that bipolar patients always sleep less wasn't the case with some. It's just a lead-in for me to build on what you and I were talking about earlier. We find different features among subtypes. Those eventually may actually be predictors for the different treatment responses. The question that was just asked back here, some bipolars may need, quote, stimulating medications. Some may actually need the opposite. And what we really need is that biomarker story to give the prediction coupled with treatment outcome studies in large samples. And you're building toward that. And in your comment about how do we manage to do this in a large framework with longitudinal studies is really the name of the game.
And you know what excites me is Nora Volkov, who's the head of NIDA, is using our ActiWatches, you know, and she's doing a study of PET. She thinks these systems have to do with dopaminergic pathways of reward and circadian rhythms. So it's very interesting that we may be able to get down to the biology of these systems through the PET studies, but also studies of animals. There's large studies with actigraphy in animal models, primate models, that might help us too.
And the final point is we really should work on the collaborative networking things that you brought up, and that is a discussion later.
I'd love to do that.
Thank you.
I
So
think
we
it's
have
important.
room for a couple more questions. The lady here and then one question over there.
You mentioned clinicians that look for symptoms to persist for a while. That was how I was trained. Can you describe a manic episode that only lasts for an hour or two? Or does it look exactly the same and it's just an hour? Because I don't think I've ever seen it. Is that typically seen more in the community?
Well, you won't see it clinically because what we're describing is in children. Well, we'll say, was there a change in mood? They'll be very active. Now, an hour, I think that's kind of on the lower end. You want to see something that's a few hours or that lasts a few days. But you'll see, what we saw in adolescence is the parent or the child would describe, I felt full of energy. I didn't need sleep. I was very active. You observe them being more active. You know, they're moving around a lot. And then it kind of just stops. And so what do they do during that period? Well, I think they can get into big trouble over 24 hours with that. You know, a young woman going out, they could use drugs. You know, these things can happen even if it's short-lasted. So my point is, for diagnostic criteria, of course we have to say it lasts a certain amount. But if we want to look at phenomenology, particularly in families where we expect it to be exacerbated, I think we do want to think of these things as dimensions, rather than there's a manic episode or not. I think that's the point. So,
Thank you.
Okay, I wanted to ask, how did you choose these ActiWatches at $1,200? And
Yes.
how does that compare to the pedometers that, Blue Care Network requires their people to wear
They are? Blue Care's requiring that? Interesting.
for some groups? Yeah.
Well, it was the only act in town when we started. So what happened recently, I got sick of paying the money. So as I have a Fitbit, I talked to the companies and they wouldn't give us the data. So Jawbone and Fitbit said, you can't have the data. I wanted the data. I said, I want to look at the data. I'm working with this team of experts at Johns Hopkins who've developed statistical models to look at functional data. So we now are, this might be an advertisement, we chose to use the Gene Active devices that are $200 each. It's a company in Britain, and they actually give you all the raw data. They're very reliable and so forth and so on. So there's a number of companies now who you can buy these. Now, with respect to the pedometers, I don't know the answer to that. So it's a little bit different. They're measuring a little bit differently than I think the ActiWatches do. These are more sensitive. But I think the pedometers will do the same thing in a way, because do you really care about how many steps, absolute, or do you care about changes and increasing? So if it's that, then you probably, you don't have to worry about it. And pedometers are very cheap.
Thanks. One final question.
Yeah, hi. Thank you for that great talk. I kind of have a tongue-in-cheek kind of question, but it relates to what you said in the beginning of your presentation, that there's a high prevalence of underlying symptoms that are not picked up by primary care and in the general population that are misdiagnosed as something else, and they might be the tip of the iceberg and so on. Recently, there was an op-ed piece in the New York Times to add lithium to the water supply. I don't know if you saw that or not, but it elicited about 600 plus comments on the New York Times, and by lay people and by healthcare practitioners alike. And maybe, do you have any comments on that at all? I just wondered about that.
What's interesting to me that, people didn't understand how lithium works. It's very effective. So this is kind of the argument of going the linear, you develop a target, you develop the drug. But with lithium, that's not how most drugs were discovered, right? Lithium, though, I've been told actually shifts circadian rhythms. That's one of the big effects of lithium. So of course, putting it in the water doesn't make sense. You don't know what you're going to do to people, it's in the water. It's like
It's in some
fluoride.
water.
But the idea of being able to treat people, I think that's exactly, it's a tongue-in-cheek comment, but it's quite interesting.
So thank you very much. I know there's going to be an endless number of questions, but I'm going to have to cut us off there and move on to the next speaker. I just wanted to bring to your attention that at the break there'll be an opportunity to ask additional questions. It gives me tremendous pleasure to introduce Sebastian Zoellner, who has been with us for approximately 9 years here at the University of Michigan. We hired him as a joint recruitment with the Department of Biostatistics and the Department of Psychiatry, with the idea that it's important to establish expertise relationships between the departments. And this is something here at the Depression Center that we have been doing consistently over the years, is establish relationships and expertise in other departments. So Sebastian has been a major contributor to, or participant, not contributor, participant, active member in our work. Sebastian did his undergraduate work in Munich and came to the United States in the early 2000s, did a 3-year fellowship at the University of Chicago studying population genetics, and has been actively involved in methods development. And so he's a mathematician initially by trade, and is truly one of the smartest people that I know in terms of analyzing data and trying to figure out all these different things. And so it is truly a great pleasure to introduce Sebastian Zoellner. Thank you.
Okay, thank you. Jeez. Not entirely sure what I'm supposed to do after this introduction.
Right. Are you glad you were wait, yeah, that's good.
I already have to apologize because, as you might hear in my voice, I'm just getting over one of those magnificent colds that your kids bring home from daycare. And so I might, among other things, entertain you with a couple of loud coughs. So one of the things you might have heard in Melvin's introduction, that I came here with absolutely zero expertise in psychiatry. So whenever I try to make sense of anything psychiatric, Melvin is the go-to person for me. And there's this one story that stuck in my in my mind that I use when I try to explain to my students what bipolar is like. It's the story of a man who was really excited about having a party. So he went out and invested his last 2 paychecks into alcohol, because you can't have a good party without alcohol. So he carried barrels in his basement, boxes of high-end liquor in his basement. It was hours away till his guests are coming till he realized, damn, all the booze is in the basement. The party is supposed to be in the kitchen. Well, I got a chainsaw. Nyum, num, 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 and he cut a huge hole in the basement of his kitchen so that everybody could get fast to the booze in the basement and have the party outside and in the kitchen. That makes for a really funny story, but it also illustrates one of the really dangerous things about bipolar disorder, right? After the party was over, the man had no more money, had a huge construction problem in his kitchen, and had nothing that he could do about it immediately. So this person could have been helped a ton if any way, shape, or form, a doctor, a technological doodad, could have told him, dude, you're about to go off to the deep end. Do something before you end up there. And that's a little bit, that's of course what Kathleen talked about, and that's a little bit what I'm trying to talk about today. So this is basically just in science what I just told you about. One of the fundamental things about bipolar disorder is that it's determined by its temporal patterns, but the problem is these are not predictable. So it's not predictable when the next depression episode is going to come, when the next manic episode is going to come, or how bad is it going to be. However, both a depression and, of course, in particular mania, will create catastrophic effects for the life of the patient. So if we had any inkling in being able to to say something in the future, bad stuff is coming down the pike, we could massively improve a patient's life. And here's a picture that illustrates how difficult this is to get. So this is a measurement of PHQ-9, which is a very, very useful and very powerful self-report measurement for depression. And these are 5 random people taken from our longitudinal study. What you can see here is they are measured for roughly 70 to 90 months. And you can see how it's different patients, so they're measured for different time spans. And you can see how their PHQ shifts from one measurement to the next. And what you can clearly see is there are different types of patients here. I mean, there is the red line. And just so that you can orient yourself, a value up to 4 is what we would consider healthy. Values 0 to 4, this is just you're being frustrated by life. That happens to normal people all the time. Between 4 and I think 18 is where doctors are concerned, but may or may not do something about it, depending on the rest of your life story. And everything that's up in the 20s is really, really dangerous. So what you can see here is we have vastly different patients. We have patient red, who is almost certainly not a bipolar patient, who never ever veers into dangerous territory. The opposite is patient, I guess that's purple, who... Okay. Yeah, okay. As I said, thanks for the illustration of my psychiatric knowledge, where we sort of make regular trips into the really dangerous categories. Glad to. And there's interesting people like Mr. Orange here, who's always above what we consider healthy, but never ever moving off into the really, really high values. A statistician, of course, I can make these qualitative descriptions here, but if you want to say something with statistics, you need to boil this down into some kind of statistics, into kind of summary statistics. And the obvious easiest ones, and I like obvious easy to start out with, are sort of our classical summary statistics. So this is the same orange patient that we looked about before. And what we're now doing is we're describing this patient by 3 summary statistics. The first one is its mean, just what's the average value of his PHQ over the entire observed phase. The second is its maximum, how bad does he ever get? And the third is the variance, how fast does he switch, or how much range does he switch? And if you look at the 5 patients that you've seen before, you can actually see that they are pretty well described by that. First, the patient with really high values, with a mean of 10 and a maximum of 27 is clearly a different patient than the orange one, who has not a much lower mean, but a way different maximum, and of course a vastly different variance. And also the blue patient, who is often in the normal range, but has a really weird outlier in the 13, is again different from the other two patients that we have here at the bottom. So this is a useful way to start thinking about these patients without having to have the whole line in my mind. We've looked at other statistics too, but this is what we're talking about. So basically we're describing the trajectory based on mean, maximum, and variance. And what we're now trying to do is we're trying to find predictors. We're trying to find in the intake, in the early interviews, do we find out something about the patient that tells us something about, is this a patient who can have really high maximum PHQ? Is this a patient who may have a really extreme depression somewhere along the next multiple years? Is this a patient who's just generally more depressed along the next multiple years? So that's what we're trying to do. There's, of course, there's two types of predictors, and they've mentioned in my title. For today, I'm mostly focusing on environmental predictors. If we have time at the end, I'm going to give you a very short idea about how we're thinking about genetic predictors, but we're not quite there yet. The benefit of environmental predictors is that they're potentially sizable effect sizes. We know by now that genetics, each individual genetic variant doesn't do all that much. Easy to modify, again, is relative to genetics. If there's something in your environment that is unhealthy to you as a patient, there's a good chance we can change it. If you don't have enough activity, we can try to convince you to go to your gym. If there's something wrong with your genes, there's not a whole lot we can do. The biggest challenge always with environmental predictors is, of course, unclear causality. You may have very low activity because of your disease, or it might be a side effect of your disease, or it might have nothing to do with your disease, right? So the causality is a much harder trick often to pull off with predictors, while genetics is pretty much the opposite. It's true. Effect sizes aren't often not very big, but causality is always pretty clear. We know that your genetic variants were there before you had the disease. Okay, so of course we're not using all in the environment as predictors, because that would be A, too complicated, and B, also lead to all kinds of statistical nightmares where you're basically looking for, where you're trying to match up to a pattern, because there's always a pattern to match to, but where you basically have too many observations to be sure that this is actually statistically meaningful. For that reason, and the second problem, of course, is with a lot of environmental measures, is they're really expensive to measure. As we've just heard, environmental measures are hard, so if we want to do something where we can find tools that make their way potentially in the clinic quickly, we want to use something that's easy to measure. So for this particular reason, we went to social environment, because we have, I don't know, I have no idea how good they are, but we have a couple of tools, basic questionnaires where we can measure social support, perceived social undermining, and childhood trauma. And these are the three measurements that we're using here. So if we're just doing the naive thing, and we look, for example, at social undermining on the horizontal axis and our depression measures, and this here is in particular case average depression, we can see that in controls there is a clear decline of depressive system of, in cases there's not much of a signal, and in controls there's a decline of depressive symptoms with an increased undermining score, which I think means reduced undermining. I'm not quite sure about that. Similarly, you can see that if you're looking at evidence of childhood trauma, you can see that patients with higher evidence of childhood trauma have also more depressive symptoms in the course of their membership in the study. And similarly, that's what happened. Sorry, the axis here is mislabeled. This should be social support. This should be social support. Now everything makes sense. So with increased social support, you have, I was just going, what's going on here? With increased social support, you have decreased depressive symptoms. So this is what anybody would expect. Better social support means less depressed. Similarly, more childhood trauma, more depression, and more social undermining also means more depression. You can see that only in social undermining do you see an effect in cases as well, and controls as well. Social undermining, if you feel particularly social undermined, again, even nominatively healthy people will feel more depressed from that. But there's sort of a problem with this very simple analysis. The first one is, if you look at a patient and you measure their social undermining, their social support, and their childhood trauma, those are not independent measures, right? Somebody who has a lot of social childhood trauma has a harder time trusting people, so he will feel more undermined by his environment. And he might have a harder time building a solid net of social support as well. So I might have just shown you 3 times the same thing. And more importantly, we have no idea what's really in the driver's seat here. These are just sort of 3 observations, and how they fit together is not clear. So in order to get a better picture how it fits together, we jointly analyzed them. We sort of said, if we take all of them together, how well do they predict what's going on? And so in statistical details, we applied a joint multiple regression. And what we did here, because one of the fundamental problems is that all these measurements from questionnaires are, of course, in no nice distribution shape. So we did a rank normalization. After that, we considered 3 major covariates. The first is obviously affection status. The second one is age of onset. And the last one is gender, which had a surprisingly large effect on all these measurements. And we regressed them out. And on the and then we used the residuals, which basically is we got rid of all of these covariates, and what was left in the data, we applied our model to. And these were the effect sizes that we observed. So these are relatively similar to what you have seen. So you can think of these as the blue bars describe the cases, the red bars describe the controls, and the larger blue bar goes up means more social support means more depression. Blue bar goes down means more social support is less depression. So this recaptures what I've showed you before in the linear graphs, that if you have more social support, you have less depression. If you have more undermining, you have more average depression, both in cases and in controls, while childhood trauma has little, may slightly reduce your depression in controls, but certainly increases your depression in cases. If you apply a statistical test to that, you will see that the effect for social support goes away, and the only thing that remains for childhood trauma is that there's a difference between cases and controls. While undermining is highly significant as a finding, as a predictor of depression. And that's just as a reminder. These are measures we're taking at uptake. This is for the mean for the next multiple years. Similarly, we get very similar results if we're looking at maximum and at variance of depression. In all of 3 of these cases, undermining stays as the major predictor, and in most of those we see that in cases there's a substantially different effect of childhood trauma, it's significantly higher effect of childhood trauma than in controls. So, yeah, this is just a summary of the data. So does that make sense? Why would anybody think that undermining matters more than, for example, how many friends you have? But if you think about it, undermining, this is not really actually undermining. This is perceived undermining, because we're asking the patient himself about how does he feel about his social environment. If a patient, assume a patient who has a great list of friends, but he thinks they are lying behind his back all the time, they are being mean to him, they are not inviting him to their best parties. How much does it matter does he has a net of great friends? It doesn't help at all. He feels like he's left out in the cold by his friends, and if at all, that makes his life harder rather than easier. So it makes a lot of sense that undermining asks directly how the patient feels about his social environment, while all the other things are kind of factual measurements. But as we're dealing here about the effect on the on the patient's psyche, his own perception is almost certainly more important than facts. Okay, so the next question that is of course interesting here is, what's going on? Is it just that if you're depressed, you're more likely to perceive that people are undermining you, and that's where the correlation comes from? Or you could just as well imagine the opposite. you're undermined, you feel that people are talking wrong about your back. Does that affect your depression? The least interesting, but unfortunately in statistics always also a possibility, is that there's just something that we haven't measured or not considered in our model. And that increases both depression and undermining, but has nothing directly to do with what we're talking about. And this is important because if we have this model, that means if with therapy we could reduce the perceived undermining, we may actually interstate here and reduce the depression. So to address that problem, and this is the type of problem that you can only address with longitudinal data. There's no way, if you have a cross-sectional study, you can write down the question and you can stare at the question for a while. There's no way you can solve it. But here we have the big benefit that we have longitudinal data. We are measuring undermining once every year, so if you can sort of see the yellow arrows here are the measurements of undermining, while our PHQ-9 measures or depression measures are done every 2 months, indicated by these white arrows here. So what we're now looking at is, does this measure up here of undermining predict us this measure here of depression? And because we don't just want to deal with the autocorrelation, we're of course controlling for the value of depression here. If that is true, that means social undermining now predicts future depression. The opposite here is of course this. Over here, if we're looking at our last measure of depression right before the measure of undermining. Again, we're correcting for the last measure of undermining because again, these are autocorrelated. And if there's something more complicated going on, we don't want this to mess up our measurements. So we're testing for whether there's a significant correlation here. The answer is unfortunately it's both. Fortunately or fortunately. If you feel more undermined, your depression goes up. If you feel more depressed, your future undermining goes up. This is not, unfortunately, easy answers are hard to come by in biology. This is one of those many cases. So what we have here is essentially a vicious circle. More depression leads to more social undermining, which leads in turn to more depression. But this is still very nice as a result. This is not nice for the patients, obviously. First, this means that if you manage with therapy to intercede here and to reduce the perception of social undermining, because remember, all what matters here is the perception of the patient. If the patient manages to focus on people being nice to him, people being supportive, we may well be able to take out that arrow at the bottom, and patients may improve. The second thing that's also very interesting here, that means if I measure the undermining in my patient right now, that tells me quite a bit about where his depression is going to go in the next couple of months, which is also by itself extremely useful. If you have a highly violent patient, I would consider adding that questionnaire to your normal office visits. It's not a ton of work to fill out, and you get a good idea about what will be happening before I see you the next time. Okay, so this is a little bit about how we're thinking of analyzing the environmental data. Now, of course, we're also interested in analyzing the genetic data. And as I mentioned before, what we're really interested in is saying something about variants that predict the genetics trajectory, which helps us because causality is clear, and that can indicate where new drugs can be developed, or even just how the whole process works, right? Because we're still working at basic definitions of the disease because we have no idea what the biology is. Basically, this is just descriptive. We're at the level of a normal doctor who says the patient has fever, and we'd like to get beyond that. So the problem with genetics is always it's a numbers game. So here Melvin and I have the benefit of being involved in a large sequencing study that has recently been funded to sequence somewhere between 14 and 24,000 people genome-wide, half of them bipolar patients and half of them controls. And this is the type of sample sizes you do need to identify variants that affect bipolar disorder, because as I told you, those variants don't do a whole lot by themselves. But of course, if you need to sample 15, 20,000 people, there is no money pot in the world big enough that allows you to get the detailed amount of phenotypic information that we have here. So the plan is that we're using this to identify interesting variants and then see in our data, when we don't have to focus on the ginormity of the genome anymore, when we just have to focus on variants that we already know do something with bipolar disorder, figure out, can they predict something in our patients? So this is the initial analysis of 1/10 of the planned sample. And in the initial analysis, you can just see that there's nothing much to see. So for those of you who have not seen these types of analyses, this is each one of these numbers here represents one of the chromosomes, and each one of these points represents a test for association. So the higher a point, the more evidence for association is there. A point needs to be on the other side of that red line for us to start being excited. So as you can see, it's 1,600 individuals. There's nothing here to be excited about. What is, however, positive is that if we look at all the variants that we already know about for bipolar disorder, they all show up. They are all in the slightly upper end, and they all give us the same result that we think we already know. And for something as complicated as bipolar genetics, that's already good. I think I'm going to skip over that. And just to summarize that also, if we're doing genetics, there's a lot of challenges that we have to worry about. In particular, if you want to analyze and store 20,000 whole genomes, you have huge computational challenges. We're going to need somewhere on the order of 3 million gigabytes of hard disk space to analyze that amount of data. That is going to use, among other things, a lot of electricity. It does. Electricity is suddenly the 100,000 per month. We also have some more interesting questions that we want to figure out how we actually understand genes. Because we have a very good idea how the part that produces proteins works. We're starting to have a better idea about how that part is switched on and off and how that's regulated. And that's where the strength of this particular data set is, because many other data sets have looked only at the part that creates the protein. But we're getting more and more the idea that basically all the action is in the regulation. And but that is not as well understood. So figuring out how to combine the right regulatory systems and to define a gene well is going to be a real challenge in this data. So this is sort of one of the major things I'm really excited about to be working on the next couple of years. So just to sum up, I've sort of showed you a little bit that describing the trajectory of a disease as complex as bipolar disorder is very complicated and will automatically always result in a loss of information. There's no way of combining such a complicated trajectory, such a complicated history, without either using the whole history or losing information. But I've also shown you that even using relatively simple measures, we can boil down important aspects of the data, and we can actually find in our longitudinal data strong predictors for these aspects that predict the trajectory of the patient over multiple years. In particular, we're excited here about undermining, which I believe can really result in improvement of therapy, both because it, as somebody who doesn't know anything about it, feels very targetable for a psychotherapy standpoint, and is something that you can measure relatively easy and helps you quite a bit about predicting where your patient will go in the next couple of months. And with this, I would like to thank my collaborators here. All the work that I've shown you, I'm just talking about it. Zongshan has done the actual analysis and has done the actual work with the help of Chris Russell and An Tuong. Melvin McInnis, as I said, provides me with sanity, at least when it comes to psychiatric traits. Gloria provides me with sanity with everything else. And on the other hand, you can see all the people who are working on the sequencing data that I've shown you. And I'm happy to take questions.
Thank you very much, Sebastian. I think that one of the best comments that I heard from one of my previous mentors, Ray DiPaolo, he says, the problem with bipolar disorder is that it's 80% genetic and 80% environmental. And I think that this, exemplifies that the interaction between between genes and what happens over the course of time is really, really important. So I want to open it up for questions.
So how do you determine what is paranoia and what is underlying? I mean, where did you put that line where you consider one paranoia and the other underlying?
My personal case, I used a score from a specific questionnaire that's designed to measure undermining. So it's well possible that some of the things I'm finding here is comorbidity with paranoia. But remember, what I find is that undermining is not very consistent. It's of course autocorrelated from year to year, but it's not like people score the same on every time across the score. And more recent scores tell you more about your next PHQs than older scores.
So paranoia is more genetic and underlying is more environmentally.
I'm no idea. It's well possible that the likelihood to perceive undermining is quite genetic. I would not be surprised if anybody finds that out.
But that's an excellent question, because undermining in this context is there's a questionnaire of social support and undermining, and so undermining is defined in this context as the perceived activity in your environment of people doing things to specifically to inhibit your progress or your success. So someone says, you need to write this report, but doesn't give you any pencils, or there's no pencil sharpener. I mean, it's really kind of a naive kind of way of thinking, but that's really what it is. And so I'm asked to do all these things, but I don't get the support and the things that I need to do it. That's really what it's capturing here. So it's not paranoia in the sense that people are kind of out to get me in that sort of abnormal psychotic, but it's sort of normal day-to-day things that people interfering with one's capacity to succeed. It's an excellent question. As you were presenting it, I thought, I'm glad this question came up because we needed to bring it up. Shervin.
Thank you for a wonderful presentation. I think in 2000, Baumeister published a paper that the effect of bad is stronger than the effect of good. That when you control two similar constructs, which one is positive, one is negative, the positive in many cases goes away and the negative stays. And Neil Krauss at the School of Public Health publishes on undermining and shows that when you control the positive effect of social support, the social undermining is stronger, stays in the model.
No, that's what I did, right? I had the social
yes,
support
absolutely,
model.
exactly, supporting your finding.
This is actually maybe a question directed at you, Kathleen, and or at the really star geneticists in a hardcore way. One of the things that I think it's time for us to start learning is that we need to get rid of the word versus in our head, because it is almost always interactive. And now in the era of epigenetics, you asked a wonderful question, but the events in our lives actually start influencing gene expression and changing the different patterns that we have. And that's what's going to make things so complicated for people like Sebastian and the whole network of people who are struggling with this. But we'll get there. We are actually making progress, and we are going to get there.
If you look at oncogenes in cancer research, it's very similar. I mean, absolutely. So if you could do the same thing, the same model, I think you could go far.
I wanted to ask a question about the controls. We kind of know how the bipolar people are chosen, but is there anything special about the controls? Are they family members, or are they in the same age range and students, or how are the controls chosen?
We controlled for age and gender. Controls are people who come into the study who want to help, who don't have bipolar disorder. So there's several, so they're not picked by any specific epidemiological criteria. But I mean, I want to point out the choice of controls really doesn't matter much for my conclusions here, right? Because I'm not comparing cases and controls. I'm comparing the idea that undermining has an effect on future depression, or undermining has no effect on future depression, right? The fact that I also see that in controls may have something to do with my choice of controls. Sure. And that may for example not be true if you pick super controls, right? If you pick controls that are screened for not having a psychiatric disease at all 5 generations back, those might not have this correlation.
So I have a question, Sebastian. Then, in the context of, I think you pointed out, with social support that many friends does not counter the effect of the undermining. Now, but would there be a strategy to look at the data to see, that so that if you just look at the balance between support and undermining, is it your point that they don't outweigh each other, so that enough social support will not overcome the undermining that the individual.
Social support seems to have the weakest effect of everything. But I mean, the problem here that you're having is that you will have a hard time finding people with a lot of social support and really high undermining, right? Because those people tend to be more optimistic about their social environment. So that's part of the problem of this analysis, that these things are not independent. But in this analysis, anything that you can learn from social support, you can learn better from undermining.
Okay. Second question I had, I'm just seeing if anybody wants to ask a question, we're going to break in just a minute. But the question I had is, did the undermining at the intake predict the undermining at year 1? Yeah, we did. Okay. Was it unchanged or?
No, it's changed.
It was changed. Yeah,
it's
up or
changed.
down?
I looked at the autocorrelation. I didn't look at.
Got it.
There was a direction. I didn't expect the directional effect, so I didn't look for
Okay.
I just want to suggest something about the connection between social supports and undermining. Those with a lot of social supports have very good social skills. And those with perceived undermining, sometimes they are at fault in what they're projecting onto another person as being undermining. So I think that's a connection there.
I'm not sure I would use the word fault, but they are certainly contributing.
Well, they're not seeing their own contribution.
Yeah, they're certainly contributing to their own problems. Yes.
Right.
Yeah.
And for the treatment trial individuals amongst the team, this is again something that we are looking at to try to identify intervention strategies, and so that what we're about is to sort of say, well, how can you identify individuals that are at risk for further episodes, and so that the ability or the report of social undermining at any given time suggests a risk group, if you will, of individuals that could be focused on.
I would just take that that data at every visit, and if you see a sudden spike in undermining, I would go back and talk to the patient and see if you might want to preactively do a little bit about the medication, or at least get him to call in 2 or 3 times in the next month or 2.
Yeah. We have questions in the back here, sir.
I just had one quick question about the time axis. On the first slide, your axis is fairly coarse. It's in months. And these conditions, very often, you'll oscillate on a much faster time scale than that. Is that a limitation of your interpretation?
Absolutely. As I said, that's why it's so nice that this actually seems to affect multiple months in the future. That it doesn't just seem to affect what happens the next couple of days, but that actually what happens multiple months in the future. There may be a lot stronger effects a couple of days in the future, those I simply couldn't tell you about. It's impossible to get weekly data from patients over the course of 10 years.
Well, with that, I thank you very much, and I think we're going to have a break right now, and so the refreshments in the lobby.
Yes, very much so.
Are you enjoying learning?
I was talking to my daughter, the one who writes well in Vermont about that just yesterday, and she had one small suggestion. I had a couple, but I wanted to discuss it with her. But if you wanted to mention it, feel free. Yes, I wanted you to know that you can.
Can I point to you?
Sure. Life has been so crazy for me.
Well, that's the problem is that it has been co-opted. Okay, so we've been co-opted doing very helpful and I really appreciate you have a very localized study after study
You didn't jump out of an airplane.
If you've never been evaluated by use of this part of the sheet, they are super quick. They like so cat got fit with proper bites. How you do this? How you can sleep with your mouth open and have apnea and have bruxism? Yeah. He does it all. At certain times. Well, so there's the thing is like his mouth. He's not perfect. Nobody's perfect. So different phases. He'll wake up, roll over, do something else, and then he's grinding while he's still breathing. It's just it's a hot mess, but he's doing tons better with sleep. Well, intelligence. How old? There's like a few years for him. I know issues that she said one day they walked into the chef's first and the parents were sweeping. They got whole pictures and the three of them.
You know, we do cortisol 4 times a day on all these people. We just haven't had money to do it.
Really quiet, I'm sure, when they were doing it.
So,
Yes, right. Good seeing you. Anyway.
So I'm asking if I know rhythm.
yes. Because the teenage, it shifts, and then we make them go to school. It's, you know, we just did this analysis of this, of the federal data. Huge. We could offset, I would, 40% of depression in adolescents by starting them after 8 o'clock. I mean, it's crazy. We can't get people to do it. School districts won't do it.
Because I went
It's the school buses, it's the parents, not the anyways. Yeah, and those kids get up later anyway. Right, right. Well, they did it in Minnesota. Minnesota did it. We did it in Cheshire, Connecticut, but then the parents fought it and we stopped. They're trying to do it in Virginia, and the school board voted against it. Right. I agree with you. So maybe that's the thing we'll take on. Hi, Kathleen.
Hi,
Nice to meet
I'm
you.
Julie Frick, and I'm a 40-year clinician working with bipolar and major depression and anxiety. And I was so excited to hear you talk about this energy because there's a separation between my clients who are clinically depressed because they're going through something in their family or whatever, and then there's this. And I say to my clients, you have to think about this as, I wish it were called energy disease.
It is. It's an energy disease. This is what we just talked about. They don't lose. You know what's interesting? They don't lose pleasure.
It's just you don't have enough energy for
They
it.
just don't have enough energy, so they can, they still, it's not, but some of them it gets so bad because they can't move. And I think it's a different mechanism. I think it's, I think of immune, you think of immune changes. That's what it is.
Well, I really want to encourage you to keep on that way because I think that's part of the stigma and confusion is that we're combining things that aren't meant to be combined. Yes, exactly. That's what it's like.
Right. So and there's some good data on that now from the right.
Well, that's the analogy I use, that it's like having mononucleosis and you just can't. Thank you.
Yeah, thank you. That's I appreciate it because it means we need to keep on. Thank you. My goodness. How much did I spill? Thank you. We had an accident. Oh, bad. You need something, don't you? You need a towel. You need. Melvin, just throw that away. I grab some paper towels, right?
Go ahead and grab some.
Should we just let, I'll just finish the question and then. No, it's not behind us. What happens is it changes on the road. So they will say their families want, there's a very famous family married with a spouses, you know, and they get better, that will change it. So it's a perception. It's what he said. I mean, I don't know it changes. Yeah, absolutely. Getting get up at the same time. You gotta get somebody other than they are totally helpless. They are totally helpless up there. Here. Oh, we did. I mean, they're totally helpless. Did you get it on any of your? May I take a look at your wonderful little wrist fun thing there? Did you see Time Magazine too? They had this picture of this whole thing from here to here on the front cover. So you're very Time Magazine. Are you happy with it? Oh yeah, I just printed. No, these are work. Feedback. Yes, this is just. What do I get? Online. And so I've only had 2 steps to make. Well, what are you gonna do? What are you gonna do? I mean, what do you? Yeah, you read this. Well, she was in line first. Okay, I'm sorry. The mess. I thought you were starting the mess, honey. Sorry. Look at us. This is good. Thank you. It was the water slide. I don't know what happened. I was standing back here and all of a sudden somebody said, look. At least it's not red wine.
I'm going to.
Yeah, she'll always make it come back, you know.
Oh, radioactive? Or
That's okay. Don't worry about it. That's right. Okay. Yes, what do you do? I really want to see. It's like
yeah, I'm going to be the speaker.
I think it's just the inspiration in advertising. And then stop images.
I just want to welcome everyone back to the final session. So, in our initial Prechter lectureship, we had one speaker who came in and spoke with us, and then I think about 3 years ago, we decided that we would have one of our local scientists present on some of our data, and then I would summarize the information, or not summarize, but sort of give an update on the Prechter projects that are ongoing. But before I start on my talk, and people are wondering why the bees, well, I'll explain that in a moment. I have a very special announcement to make, and that is a unique contribution, collaboration, and establishment of support here for the bipolar research that's ongoing here at the University of Michigan in collaboration with the Heinz C. Prechter Fund. And that is that this spring, or the end of June, we received a most noble gift from the Richard Tam Foundation. And I'm here today to announce to you that we received a gift from the Tam Foundation for $350,000 to support research in bipolar disorder in collaboration with the Prechter Fund. And I want to introduce Judy Sargent, who's the president of the Richard Tam Foundation, and thank her for her work in supporting us. And we're very excited to have the Tam Foundation partner with us, partner with the Prechter Fund, partner with the University of Michigan, and be a player in the team that is working on finding solutions for bipolar disorder. And we're so grateful for everything, and so grateful for your dedication and your support for us. We're very, very happy about that. And so with that, I also want to start and talk about, why this slide. So over the weekend, as I was putting together, in PowerPoint, they have these title slides. And so I got some of these title slides, and I was putting out, identifying the title slide. And I kept getting iterations of title slides that were from the Michigan, the victors from Michigan. And so I thought, well, I like the bees because bees are busy and they do things. And there's a reason why we have, busy as a bee. And so that, and bees do good things, and bees are very important. And I have a story that one of our patients called up one time, and he had heard the concerns about the bees, and he had concerns that the bee population in the United States is dropping, and so that he said, so he called up, and the individual in our intake on the intake line said, this person, then all of a sudden they went off about bees. And I said, well, it's just the concern about the national situation with bees. And so when I saw this, I just thought these are very earnest and serious-looking people here working on bees, and you can see the look in the guy's eye there. And this is, it's the equivalent of the scientists looking at the data for bipolar disorder. So I want to point out that the longitudinal study is really possible because of the dedication of so many. First and foremost, are the individuals with bipolar disorder that are contributing their personal data and all of their information to us. And we have literally hundreds of donors who are providing support at various different levels, including financial resources, including time, and then just with the word getting out there. One of our patients said, spontaneously to her neighbor, who happened to be a researcher here at the University of Michigan, she said, I participate in the Prechter study at the University of Michigan, and because I'm participating in research, I just do so much better. And it's that kind of word and that kind of enthusiasm that helps us tremendously. Jennifer and others there, or it was Valerie, Valerie's here, she got an email, or we got to one of the participants in our cell phone project talking about how wonderful it was to be participating in a study that's right on the vanguard of research, and telling her what a pleasure it is. And of course, there's so many people that are involved here: the leadership of the department, the Depression Center, the development officers at the University of Michigan development officers, and of course Wally Prechter here stands out, and I just have to say the name. Because of she needs no introduction, and she just stands out there. The other points I want to point out, or the other things here, are the Heinz Prechter Research Fund, the Stephen Schwarzberg Fund, the Depression Center supported us, the Richard Tam Foundation, NIMH, the American Society of Suicide Prevention, and the Thomas and Nancy Upjohn Woodworth Professorship has also contributed to this. And so the genetics repository is a very complex repository, and we're gathering a wide degree or wide variety of data and information. So we have longitudinal data from our individuals, we have genetic data, stem cell data, nutrition data, electrophysiological data, imaging data, and now voice data. And so when I talk to the people in information technology, he says, son of a gun, McInnis, that's a lot of data. Then I don't think that this one database is going to manage it. And so what it is that we're kind of a federated data system. And so we have knowledge of all the data points that are here and there and centered around the Prechter repository. But all of the roads and everything leads right into the repository. And so here are the numbers. I'm going to draw your attention to the top line there, the University of Michigan, around 1,049 individuals in our longitudinal study. But more importantly, I want to demonstrate to you the amount of work that is being done. And so this time 5 years ago, we had around 8 individuals per week that were coming to the clinic, and now we're doubling that effectively with 16 individuals that are coming. So there's a lot of work, a lot of logistics that goes around this. And so a new patient or new person coming into the study mortgages a whole day coming in. Then sometimes we split it into a day and a half or 2 half days, or sometimes it takes a little longer. So there's an enormous amount of work behind the project. And so the ongoing monitoring, the initial visit is very involved, and so it takes around 3 hours for this neuropsych testing, which is extensive testing in IQ testing, and being able to do these intolerable exercises that the neuropsychologists have the individuals do, and these go, no-go things. The diagnostic interview, the genetics and biomarkers, and the questionnaires, and then we have the individuals follow up on a bi-monthly basis with these questionnaires. Now the challenge that we have had with that is that getting paper things in is uses the U.S. mail and all that. So we thought, okay, well, we're going to use technology. So it turns out that technology is not necessarily a strength, because people will stick it online, the phone rings, and they go do something else or whatever. And so, oh, well, I'll continue with that later. And then a month or 2 passes where they just, they see the piece of paper on their desk. Oh, I'll fill it in, and they just get it in the post. So, so technology is great, but it's not going to be the end-all and be-all. So this just gives you a distribution. It's got 960 individuals that we're reporting here, but around half of them have the classic bipolar 1 disorder. Around 10% have bipolar 2. 5% or so have the bipolar not otherwise specified, and we have around 236 healthy controls. The interesting thing about healthy controls, so a healthy control in this instance is someone who doesn't have the disorder. However, many of them will call in and they will say, I want to participate as a control, and you ask, well, good, great, wonderful, and they come in and they've got depression or something else is going on. So the whole concept of a healthy control is rather dynamic because, in fact, of our healthy controls, we have the population frequency of bipolar 1 disorder, major depression, and others. So that the healthy controls over time, we find all kinds of problems with them. Or not all kinds, but we frequently find problems. So I wanted to, just to emphasize for you, I showed you the federated data system. And so we have a number of projects that are booting off, dependent on, interacting with the longitudinal study here that we are doing. So these studies here that we're looking at, we would not be able to do these studies were it not for the presence of the Prechter Bipolar Longitudinal Study, because we invite these individuals to participate in this study, and we call them up, or we have a particular strategy that we use presently. And so we identify the criteria for a new study, and then we send out an email to the individuals that we believe are going to qualify. And it's up to them to contact these other studies and say, you know, I heard about this specific study and I'm interested in it. So we've done this on a number of times, on a number of occasions, and had a rather resounding effect in the sense that investigators have called up and said, "Thank you very much. I filled my study in a record period of time." To look at, for example, the nutrition study here with Simon Evans, which is a study looking at how individuals' diets are, and so they have a particular protocol that they go through to measure what they eat, and then they come in and Dr. Evans does a series of tests and blood tests and so on. And so it's a way to really get a clear picture of what's going on in the blood and the metabolomic pattern of the individual. He filled up the study early. He was at about 125% of his recruitment. And so this is a study that's funded by the NIH as a training study for him. So many of these studies are successful because of the fact that we have this base Prechter study that are going on. So what is our approach, and what are we looking at? So we think that bipolar disorder is so much more than just a cross-sectional diagnosis of bipolar disorder. So we're looking at everything from the disease to the categorical diagnostic features, and we're looking at medical outcomes. The process of studying bipolar disorder involves the longitudinal course of the illness as much as just saying, bless you, my son, you have bipolar disorder by a one-off, 20, 30 years ago, we thought we were pretty smart and we could just make the diagnosis right then and there with an interview, but we've now learned that we have to follow people over time. There are behaviors that drive features to, that exacerbate bipolar disorder. There are dimensions and personalities. So everybody has a personality. Everybody has a base kind of set of features that drives the impulsivity of the illness, that drives features that lead to things like suicidal behavior. Them in and of themselves are not necessarily the cause of it, but they're contributing to it. We all have a life story. We all have things that happen to us. And so Dr. Zoellner just showed you how features of the life story of the individual are impacting the outcome. And of course, circadian is very important, and Dr. Merikangas talked about that. Now, equally important are neuropsychiatry, and that is the cognitive functioning, our memory, our motor control, our executive functioning, how well you do on these particular tests. And it turns out that there may be an influence of bipolar disorder with aging on these executive functioning, and we've published on that in the past year. So this is a really messy slide, and I love to show it because it's so messy. And it's I call it, the Dow Jones of bipolar disorder. Things are going, up and down all the time, and how do we make sense of it? And so you think, and you think of these stockbrokers looking at these numbers going across their screen. You ask yourself, gosh, that's complicated. And but I want to point out that there are patterns of the disorder that we're seeing there. And these are 40 individuals of roughly 500, so it would be a real mess if I showed everything. But this is what Sebastian is charged to do, is to sort of start parsing out the patterns of these illnesses, of these patterns of the symptom severity over time, correlate them using statistical methods and mathematical methods to start to make sense of it. And so So, you know, if you look at the Dow Jones and you think, well, okay, well, it's a normal distribution of all of the prices of the stocks. You know, you can look at it like that. But we all know that there are, you know, there's variability in the types of stocks and so on and so forth. And so the question that I've challenged them with is that how can we start making sense of these patterns? Similar to what we did with biostatistics, when we were faced with a situation to start making sense of cellular models, and I spoke with Sue O'Shea about 5 years or so ago, and we embarked on a study with pluripotent stem cells. And so stem cells is a tool that we can use. We can get skin from the individual with bipolar disorder and make them grow into these primordial early stage cells that can subsequently grow into neurons. And these neurons reflect the genetic makeup of the brain cells or the nerve cells in the brain and behave, we believe, in a manner that would be reflective of what the nerve cells are doing. So we have 33 participants. And again, these are individuals from our longitudinal study. We call them up and say, gosh, son of a gun, are you guys doing this? That is great, and I'm willing to participate. So they come in, and they mortgage, again, you know, a half a day or day, and they let us take a sample of skin, and Sue or someone from her lab goes over to the hospital and, you know, gets the sample, and then they go back to their basic laboratory, and they set it up, and they start working on the setting up of these lines. And so this is really what how it's done. So a patient, you know, they grow these cells into these induced pluripotent stem cells that become neurons. And then there are a whole host of opportunities that we can use, from testing new drugs, testing lithium, testing ketamine, just to name a few that we've been participating in. Now, the other thing that they do is that they sell their artwork at art fair. So if you look at these cells, there is beauty in it. And just to go in and see it, and to see that these are nerve cells that are derived from someone, you know, in our clinic, and it's, you know, out and about there. And they demonstrate that they've got all of the primordial layers of the developmental layers that are there, the mesoderm, endoderm, and ectoderm. And these can grow into stem cells that have all the appropriate markers that say yes, or they can grow into nerve cells that have all the appropriate markers and say yes, we are nerve cells. And it is truly, truly amazing. I'm hoping, yes. So I want to give you an example of how these things fire. And so you're looking at here cells that are growing in a dish. And these are nerve cells that are all growing together. And so that's, think of it as an old-fashioned telephone network. And so that, you know, in the countryside where you rang up from Central and it went, you know, to all of the farms in the valley. And so this is what's being seen. So you can see then they start stimulating it, and so all of a sudden everything just all the lines light up, and this is what they see in the laboratory. And so what they can do with that then is measure the intensities of the dye change that reflects how the cells are firing. And so it's all about the cells firing. So you have here an individual with bipolar disorder in the red and a control, and they're grown in the absence of lithium in these two situations and in the presence of lithium. Okay? So you can see in the absence of lithium, there's a significant difference in the height of this column compared to the controls. Now, when the individuals with bipolar disorder are grown in the presence of lithium, the difference is less significant. It's not significant. It normalizes. So this is prima facie evidence that we're able to demonstrate that a cell or a set of cells grown from an individual with bipolar disorder that are reflective of what's going on in one's brain, that they're more reactive compared to the individuals that don't have bipolar disorder, and they're calmed by co-culturing them with lithium. Now, this is very significant, because it offers a particular model by which to study bipolar disorder in the cell, in the actual laboratory, so we can test this. The challenging thing that we have is that these are time-consuming experiment. So it takes about 12 weeks to get those cells up to that particular time period, and then you grow them together with lithium for a day and do the measure. And there's all kinds of finicky things that that can happen. This is a typo. That should be aggregates of fibroblasts here. And so we're looking at a particular behavior. This is from Sue O'Shea's lab, and Monica, I think this is your work that you just hot off the other president, Monica Bain here, that's been doing this. So you're looking at clumps of cells, and there's little red things that come up out of them. And they're a particular feature, the cilia of the cells, and so it's representative of a particular chemical or neurochemical feature of the cell, or neuroanatomical and chemical feature of the cell. And so they count the number of little red arms that come out of them, and so that is a measure of the health and functioning of the cell. And this is put in 3 dimensions here, just to show that these are clumps of cells that are growing together. So the first stage in doing this is to do it at the level of individuals, and we anticipate as time goes on that we'll be able to do this in a more automatic method. And so going forward, we have 15 individuals of our 33 that have been reprogrammed into stem cells. We want to get to approximately 60. Now, this is a smaller number compared to the larger things that the geneticists want to do in their bigger schema of things, but 60 represents actually a very large number of stem cells that are growing, and it's a major operation. So we're going to be testing medications, and so we think going forward that this is going to be a method for personalized medicine, so that when you develop an illness, a chronic illness at an early age, why would you not want to have a system to be able to test what would be the most effective intervention? Why would you not want to have a system that would test what the outcomes are going to be? Could you predict if the individual is going to have cardiovascular disease? We're talking about mania and cardiovascular disease being a very serious problem. So if we could know in advance what the risks are, it would be extremely helpful in guiding the treatment. So it's a personalized way of approaching medicine. And so then looking at animal models, and so we can knock out genes and make all kinds of alterations and different things to fuss with. So the next thing I wanted to talk about is that some of you have heard we've been very fortunate to have some media coverage of what we call Priori. And Priori stands for the program called Predicting Individual Outcomes for Rapid Intervention. And Priori assumes that there are changes in the way that speech sounds in the individual with a mood disorder, and that change in the sound and the acoustic features of the speech will predict a health state change. And so the hypothesis is that speech measures our internal emotional and mood and affective states, and so these features of the speech, the acoustics, predict state changes. And the advantage of this is that it can be collected remotely, relatively cheaply, using a mobile phone. And so I'm a participant in the study, and so my phone captures my speech when I talk on the phone to anyone, and it zips it up at the end of the day and sends it to the computer science department for analysis. And so what is the inspiration of this? Well, anyone who has dealt with someone with bipolar disorder and knows them reasonably well will be able to say, something was going wrong. I could hear it in their voice. So you talk to any parent that's got a child in college, and they will tell you the story. He said, listen, I talked to them last weekend. Something's going on. I don't know what it is, but they didn't sound right, And so, kids, they do their best. Oh, yeah, Mom, everything's good, fine, and all that. So the kids learn this game, the sound better. And there's a couple of interesting stories around that. But this is what the program really does, is that it uses a phone like this, collects the calls. The little key here, it just represents security. And it took us about a year and a half to get all the security issues sorted out for this. And so we're looking at, then it comes to the central state here, the central computers. We're looking at feature extraction. We have individuals, Jennifer Montgomery collects the YMARS, the Young Mania Rating Scale, and the Depression Scale. So this is to establish what the engineers call ground truth. And so when they first used that term, I said, what do you mean by that? And well, we want to know exactly what's going on. I said, well, that's sort of a challenge, because even though someone answers the HAM-D or the depression rating scale of a young mania, we're not necessarily clear that we have exactly the ground truth, but it's about as close as we can come. And so then we're trying to associate these features to determine whether the individual is manic or depressed. And so what happens? It's been such a wonderful experience for me because I've learned a new vocabulary. I've had to learn a new science. And I've learned such a tremendous amount in trying to understand exactly what the engineers are doing. So this is, speech, and so we're making noise, blah, blah, blah. And they have very short periods of time that they're sampling the speech. So in a 3 to 30 second interval, they're just taking these 25-second intervals there and capturing the data there. And then they measure a particular segment, and then they apply a particular statistic to this. And so I've learned about, shimmer and jitter and all these different things. I have to go and look it up, and it reminds me of a joke that I read in the New Yorker, and the joke was, do you know it, or do you Google know it? And so I have to, that's a deep joke, actually. It's a, because we always sort of pontificating about something, you just sort of Googled, and then, so I have to confess that I Google know this, but so jitter and shimmer. So if you remember, in Mr. something, Bill the Science Guy, you'd see these sinusoidal waves. And so it's either shimmer or jitter that is the deviation from that nice sinusoidal wave that you see in the old-fashioned oscilloscopes. And so there's around 52 different measures that our colleagues in computer science are analyzing and coming up with a statistic on this. And so the other point, the point here is that we're not looking at the lexical content. In other words, we don't care, what the motivations are. By I'm talking to mom, or I'm going to go shopping. It doesn't matter to them. We're just interested in the squiggly lines in the acoustics of it. So we're not looking at motivational things. We're not looking at recognizing specific words or anything like that. So our initial data are actually quite encouraging. And so what they've been able to find is that in the hypomanic episodes, the revved up periods of time, we're able to identify with a greater degree of accuracy the changes in the acoustic patterns compared to the depression. Now, this is of interest, and I was interested in what you were saying about, with the mania being more predictive and the mania associated with the energy, and that, because I think that is of considerable interest on it. But it's also the fact when someone's depressed and you talk on the phone, and you don't necessarily, the individual has a certain degree of insight that they are depressed, and so they will hide the fact that, yeah, mom, I'm fine. Oh, yeah, dear, don't worry. Oh, yeah, everything's great. And so we have just a tendency to try to conceal the fact that depression is there. Whereas in the hypomanic episode, oh, yeah, everything is great, and you just, and so the people are just really, and they just really want to, sort of exemplify the amount of energy and the things that they have. Their insight is diluted to the point that they don't appreciate the fact that they're talking a little bit charged up and over the top. So the advantage of this is that once the kinks are worked out, once the hardware is, once the programming is sorted out, and once the algorithms are written, and once everything is done, and I just want to introduce John Gideon, who's the grad student who's working at this, that this is a scalable process. The advantage of it is that all the individual has to do is to carry their phone with them. And so we don't have to have them or ask them, put on something. You don't have to, answer any questions. All you have to do is, if you will, answer the phone. Now, it's been brought to my attention that many of the teenagers are not answering the phone, if you will, very much. They're texting. And so now, John and others, they've got a program. We've got the ethical approval to count how many times the phone is fussed with. And so that every time you open your phone, what your data usage is, what your battery degradation rate is, where you're going, how many times you call your mom, sort of a thing, can be all captured, on the phone. So we're gathering a picture of what the illness, what is going on with the individual. Now, why do this? We do this because we want to be able to identify a pattern of use in the individual. And we do this because we think that if we can identify a change in the pattern of use in the individual, that will predict some change in the health state of the individual. Now, we can't know, that what's going on, but we can just say there's something going on, and something needs to be addressed. We're not at that level of that yet. We're still, John and Emily Provost and Satinder Singh are still working on the relevant parameters to really establish what we need to study. But our next phase will be to engage individuals over time in a clinical trial. Then we're talking maybe 1 to 2 years to attempt to determine whether an intervention strategy can be deployed and whether we can actually truly make a difference. Because if we can't make a difference at the level of someone's illness, then it may not be anything of relevance. But it's also been pointed out to us, and we appreciate that there are other neurological disorders, there are respiratory disorders, there are cardiovascular disorders. So someone who has congestive heart failure is a little bit congested and is short of breath, and so if they're deteriorating in their health state, there may be changes that are afoot in the way they sound, and of course someone with asthma and someone with neurological disorders and so on. These phones, they have gyrometers in them, so that if you're walking around and all of a sudden they start becoming dizzy or things like that, the phone can start picking that up, and it'll detect if there are things like that going on a regular basis. So the next decade that we're interested in, we've had a number of future is next exit. I found that on the web. I know, I Google know it. That's I Googled it. This is that Google knowing it. The New Yorker has got really brilliant ads. So where are we going to go? How are we going to go forward in engaging strategies to understand bipolar disorder better? Well, I do think it's not going to be a question of whether technology is going to be useful in research or whether technology is going to be useful in care and management of individuals with bipolar. The question is, how are we going to deploy it? How are we going to use it? What are the strengths that we're going to find? And how can we go forward, engaging technology? And I think that, we have to have some kind of a hybrid of what we have now. So for example, we could send a text, is this a good time for you to fill out your questionnaires? And you can say yes or no. If you don't respond, then we know not to bother them, because I've done this. I did this for 3 months. It was not the most wonderful thing that I did. I mean, you get this text every day, says, oh God, I got to answer this thing. So we have to figure out how we can accommodate it and align it with the individual's pattern. We have to look at, extending our collaborations around the country. And we were on a conversation with California the other day and said, listen, this, once the technology and once the infrastructure is clear, there's no reason why we can't have 100,000 individuals, capturing data, like this on them. There's no reason why these individuals could not just go to their lab or their clinic and just have their blood drawn and then the DNA sent, and and then we've got deep data on 100,000 individuals rather than the 20th century type of data on these individuals, just sort of a yes no to a series of questions. And so we hope going forward that we'll have much deeper data on them. The biology is going to be extremely exciting, and I think that it's not whether large numbers of genetic studies are important or whether we need to have the detailed on it. We need to have them both, and they're going to both inform each other. And our strategy is going to be to focus on the iPSC here for the bipolar individuals. And lastly, I think that we have to think about how we're going to impact people's lives. And so I remember, always the point that one of our participants, said, listen, because I'm participating in this research, I'm just doing so much better. Well, we want more than that. We want to figure out strategies that will engage individuals to participate in research, but also to give something back. And I just want to, thank Amy Kilborn for the discussions that we've had with her project on life goals and advertise that. And that is how we're going to use technology to help individuals establish life goals, or what they want to accomplish in the next week, the next day, and the next period of time. And the information that Sebastian and Zoellner and others are looking at with social undermining, those kinds of things can be potentially useful and integrated into life goals. And so it's not just about the research, it's not just about the care, but it's really, as we're talking about, integrating research with care, integrating research with our outreach programs. And one of the things that I think that is so wonderful about being here at the University of Michigan is that we have such a diverse set of scientific endeavors, scientists and departments and expertise in so many different arenas. So we've got a world-class and School of Engineering with biostatistics that are top of the line. We've got psychology, we've got social work, we've got health services, and that are going on in addition to the things that are going on here at within the medical school. So the reason that we are fired up about it is that Michigan is the place to do this kind of work. And so as I conclude, I again want to thank Wally Prechter for her insights and for her vision and her support over the past decade. And I thank all of you for coming here today. Thank all of you for the contributions that you've made in every way. And just coming here and hearing about our work is an extremely valuable contribution, and it's your time and your energy in that. I want to thank Judy Sargent and the Richard Tam Foundation for linking up with us and joining our forces. I want to thank Kathleen Merikangas, whom I've known for years, for coming here today. And we're energized by the energy that you have, and energized by the fact that it's the energy in these in bipolar disorder that is key, because that gives us something to work on and something to take us forward. And also thanking our staff and the researchers that have worked on the project. So I thank everyone for coming, and really thank you for making this a very successful day. And happy to entertain questions now. We can also ask Kathleen and Sebastian more. Yeah, that's right here. Or the Wally, yeah.
When you talk about the stem cells that are being bombarded with different medications, I think we talked about bombarding them with electricity, not unlike electric shocks. Will we do that?
Thank you very much. I didn't mention that, but that is something that we brought up at the last lab meeting with the stem cell group, and to think through exactly how we can physically treat the cells to either to make changes in them, but also to cause them to be more ill, and to what can reverse that. But that's a really good idea.
Dr. McInnis,
yeah.
I have a question about the Priority Project. Do you have to account for variations in individuals with mania? Is one person's mania different from another, and does that come into play?
Yes, so one person's mania is absolutely different from the other, and what we're interested in learning is whether at the individual level, and this is why the person carries the phone for 6 months or a year. So we're interested in learning their baseline, and I use the analogy that, because I grew up on a farm, and so my father emphasized for me, the importance of the sound of a well-running machine, because when something goes wrong, you hear it. And so each machine sounds different, and it has its own developmental, phase, if you will. Or the sound of an animal in distress. And so each individual has their own rhythm, if you will, of how they're going forward in life and how they sound. And so with the machine learning process, the computer technologies that John, Gideon, and others are working on here are to develop a signature for the individual. And so when the individual varies off that baseline signature, that's when we can determine that there's something that we should respond to.
Sue, are you still here? You are. You're back there. So correct me as I get this wrong, but one of the more exciting aspects of the induced pluripotent stem cell projects in my eyes is the fact that it can back it up to where things got started and then run it forward. And almost like a moving picture or like looking at a set of photographs over the span of 30, 40 years, you can sort of say, ooh, what just happened there? Did something go wrong? Is there a framework of identifying time points when the abnormality may have occurred? Those are the clues that can open the door to preventive strategies, and it may take a while, but that's actually something that sort of says not just how do we learn how to treat people more precisely now, but what could we do to sort of say this is the time point that tips off when things went sour. And I like that.
I will address that, to send that to Sue. Sounds like a 50s movie, but
if I could have my slides, please.
What's that?
If I could have my slides, please. So I would give you one example, John. I mean, that's exactly what we hope to do. And the reason that Monica is really looking at those tiny little cilia is that the bipolar cells don't have the gene that makes the cilia. So if you think about mechanism and why the patterning of the brain might be off, it's because, you know, they don't have the structural protein. So, I mean, it's a really good example, I think, of how we're trying to pinpoint where in differentiation things might go wrong. No.
Because if you think about that now, just in an imagination, and having bipolar neurons that don't have cilia, let's just say that some of us had a family history and there were time points that we know that risk occurs. So ages of onset tend to be 15 to 24 in a lot of people. We could go back and potentially look at some of those, and all of a sudden you say, this individual's at risk. We're already finding evidence that's not there of a normal developmental pattern versus this is okay. And now you start having interventions that occur not 10 years after someone has struggled with these things, but 10 months.
Yep.
And suddenly we're in a preventive strategy.
I've been very impressed with all of the planning about interventions and monitoring and everything we've just discussed. And I know it's very much down the road, but after we feel that we know more than we know now, where are we going to get the people to do the interventions?
Thank you very much. So actually, it's not all that far down the road. So we are actually in the process of preparing the ethical application to do the interventions for a number of these ideas. The other important point to stress is that this is research, and this is this is research that is really at the vanguard of knowledge. And so we don't know. We really don't know whether this is going to work or not.
I believe that you don't know.
And we would like Thank you. I'm glad I made that apparent.
Oh yeah, absolutely.
No, but so we are planning these projects, to do the interventions on it. And so what we are doing actually is that we are going to the individuals that are participants in the study and saying, this is the project that we have. Are you interested, and would you be willing to? And here are the benefits, here are the risks of participating in this project. And so we go essentially to the individuals that we're collaborating with, the bipolar, I call them participant collaborators that work with us in doing this. And our experience to date is that they've been very enthusiastic about helping us out. An individual that participates in our longitudinal study has been with the illness for 20 years. They're 40 years old, usually in late 30s, early 40s. They have had a good number of episodes themselves, 5 or 6 episodes of mania. They want to try to find some solutions to this, and they're very enthusiastic, to help.
But how do you operationalize their enthusiasm? I mean,
So
what in fact would that mean in terms of an intervention down the road?
So are you asking what would be the design of the study?
I'm asking, you elicit their help.
Yes.
And how do you operationalize their help? For example, are they going to notify you? Are they going to call other people? Are you going to have other people intervene to do help? Help means treatment. Yeah. How are you going to, who's going to be monitoring all of this?
Got it.
I love the research,
Yep.
but when we get into operationalizing it, what are we going to do?
Yep. an excellent question. There's one more question that I want to add, but I can summarize this very succinctly, I believe, with using an example of the priori. So imagine, if you will, that we have individuals, 100 individuals engaged for 2 years. And so we have their system, red, green, and yellow, or red, yellow, and green, the lights. And so if they're at a red light, said something's going on, something has to happen right now. If you're at the yellow, there's a warning sign there. That means you should see your doctor straight away. So the purpose of the study is to really just identify a likelihood of a change occurring. And so that we would say you have, a 70% chance of something going on or something happening. Please come in. Let's do an assessment, and based on the things that we can do right now, in other words, we can change your lithium, make sure that you're on target with your lithium level. We can make sure that you're sleeping better. We can make sure that we can clinically evaluate your energy and your sleep and intervene with the current medical treatments that we have at this particular moment, because we have treatments that help people by and large. It's just that they don't comply, that there's adherence issues, and there's a compliance issue, and then there's side effects. They say, I'm not going to take this because of X, Y, and Z. Yeah.
I just want to say, too, that we have over 1,000 people enrolled, and we're in touch with the entire population every 2 months. So we're actively engaging with them.
Last comment. So let's say that they're checking in with you, and I'm sure all of us here who have treated these folks who are actually great patients at times, we have a compliance issue, and many of them who would not be in a face-to-face treatment wouldn't be complying to their pharmacology at all. And so we can intervene in the way that you're discussing, but then what about the one-to-one face commitment to these folks?
Well, that's the test of the technology. The hypothesis is, can you use technology to monitor them in that, and that's part of the testing, and so that's what we want to test. We have one question here, and then one. Judy, yeah. So we'll just give Judy priority here. Yeah.
I just wanted to ask a question. With the different medications that are being tested with the stem cells that have been turned into neurons, I see that you've tested Buspar and lithium. Have you tested any other medications as well? And when you're testing them, would you test only mood stabilizers, or would you test antipsychotics or antidepressants as well?
Thank you very much. Yes, we've tested those medications. We have the cells lined up now to test ketamine, which is a newer medication that's being used for more instantaneous treatment of depression. And then, so the next phase is to systematically go through these medications that are effective and look at how they influence the cellular machinery in individuals who respond to them.
To tell how well the stem cells would be, the neurons would be reacting to these, would you be looking for those cilia that are missing on people with bipolar disorder that normal controls would have? I mean, how would you differentiate what medication would work better than another?
So what we would look at, we probably wouldn't use the cilia because that's more of a functional measure in an earlier stage cell. But I showed you the cells firing. When it's that model that we would be using more specifically to see how the cells are firing, how they're interacting with each other. Other cells, when they're treated with specific medications, they change their profile, their signature on the external side of the cell, so that there's a variability in what they have in their receptor profile. So there's a whole host of different things that we'd be looking for, but most likely there would be no one specific marker for any one particular medication, but rather a sort of a series of different things that we would be measuring and comparing them to the normal controls. Yeah.
Sorry, I have just one more question. I know that there's DNA testing out now that can say what medications would be the least problematic, the next problematic, and those you have to be the most careful because of a DNA profile for antidepressants and antipsychotics. But I don't know whether those are available for mood stabilizers.
So the tests that you're referring to are the metabolizing enzymes, so that there are people that are high, low, and medium metabolizers, and so that if you're a low metabolizer, for example, for some, then you don't need as much of an antidepressant compared to a person who's a high metabolizer because they're chewing up the medicine. But there are no tests like that that really measure the mood stabilizers, in particular lithium, because lithium is just an ion that is not metabolized by those enzymes, and likewise with for Depakote. So we have one question over here. Yeah. Yeah. Thank you, John. I did. So one question here, and then one lady in the back, and then we have reception following.
Okay, so I want to thank you so much for the day, and I've come. This is my third one that I've come to, and as a trained math person, well, I only have a bachelor's, but it's really exciting, the math ideas and the phone and pedometer sort of studies. But I just want to add into the idea mix that I don't see anything here on family systems. So I'm a child of a mom with bipolar, and also saw it in, wasn't necessarily one of them was, okay, I'm not going to go with that, sorry. But several generations, as many of us know. I don't have a diagnosis, but I have a sister who does. And it's so exciting to see the technology side applied, but nobody's trying to study what if for each of your 1,000 people, you have a roommate or a family member who lives with them who's willing to fill out that survey 4 times a day, or once a day, or once a week. Start there.
Right. Thank you for bringing that forward. We do have what's called the Family Adhesion and Cohesiveness Evaluation questionnaire, which is called FACES, and that measures, what kind of a family, the base information from the family. We haven't presented on that yet, but we do have that data. And the other thing that I think is also interesting is the experiences in close relationships, and that reflects the family patterns. So we're measuring, getting that kind of data as well. So if we can just move on to the one final. Yeah. Thank you. Yeah. Well, thank you very much, everyone, for coming, and I think we have a small reception afterwards right now. So thank you for your coming.